Good afternoon, everyone, and a very warm welcome to day two of PHARMASTEER 2023. Thank you so much, everyone, for registering and joining us today. I feel extremely proud to say that this pioneer virtual orientation program, which is organized by Department of Pharmacology and Therapeutics, HGS Medical College and KEM Hospital, Mumbai, in collaboration with AstraZeneca India, is receiving great appreciation this year as well. With that, I, Ashish Kumar Dubey, Manager, Medical Excellence from AstraZeneca, would like to welcome you all once again. I hope you guys had a great time last Saturday with the topics related to your role in the industry. And without any further ado, let us jump directly onto the first topic of the day, which is pharmacogenomics in new drug development. And for that, let me invite Dr. Nitya Vogta on stage. Dr. Nitya is professor and head Department of Clinical Pharmacology, Sage GS Medical College and KEM Hospital, Mumbai. Dr. Nitya is former editor of Journal of Postgraduate Medicine, and she is also former member of Advisory Board, British Journal of Clinical Pharmacology. Dr. Nitya is also chairman, Ethics Committee, Tata Memorial Hospital and Hinduja Hospital. With that quick brief, Dr. Nitya ma'am, over to you for steering us forward. Over to you, ma'am.
Thank you, Ashish. Good afternoon, everybody. I'll begin by talking about why we need drugs. And I think there are two fundamental reasons why we need drugs. One is because they alleviate suffering, and the second is they also help us to lead longer and more productive lives. Okay. The question is, do they really help us to lead longer and more productive lives? And I'd like to talk about an old case report which was published in The Lancet in 2006. Now, this was a full-term healthy male infant which was delivered vaginally. On day 7, there was difficulty in breastfeeding and lethargy. On day 12, there was gray skin and reduction in milk intake. And finally, on the 13th day, the neonate was dead. A post-mortem was done which showed no anatomical abnormalities. The concentration of morphine in the neonate was 70 nanograms per ml, with the normal range being 0 to 2.2 nanograms per ml. Okay. Now, you might wonder where this morphine came into this neonate. So the mother was prescribed a combination of codeine and paracetamol for her post-episiotomy pain, and she took these tablets for a total of 2 weeks. So codeine is OD methylated to morphine. When the mother was genotyped, she was found to be an ultra-rapid metabolizer. Ultra-rapid metabolizer, and the maternal grandmother was also found to be an ultra-rapid metabolizer. And the clinical picture was basically consistent with opioid toxicity leading to neonatal death. So this brings us to Sir William Osler in 1892, who said then that if it were not for the great variability among individuals, medicine might well be a science and not an art. Let's take some other examples. And this is a commonly prescribed anticoagulant, warfarin. Now, this ranks the first in terms of mention of deaths for drugs causing adverse events based on an analysis of death certificates. It ranks among the top drugs associated with hospital emergency room visits for bleeding. And even though we have the NOACs today, the minor bleeding rates in our cities has been as high as 29%, which is why warfarin continues to be used. And if you look at safety, continuing with safety, adverse reactions essentially account for 5% of all hospital admissions. They occur in 10 to 20% of hospital inpatients, and they actually cause death in 0.1% of medical and 0.01% of surgical inpatients. And there is an economic burden, depending on which part of the world you are or which part of India you are. There is an economic component, there is an economic burden of these adverse drug reactions. From safety, if you actually move to efficacy and you look at drug responses, you will find that depending on the disease that you're looking at, you have poor response anywhere ranging from 20% to 75%. For example, asthma and beta-2 agonists, the rate of poor responsiveness can be as high as 75%. And in hypertension, depending on the drug class that you're looking at, anywhere from, I think, half to 1/3 of the patients can actually be non-responders. And therefore, what we are coming to really is to the understanding that medicine, any medicine, is really personal, right? And all of us are different. So the questions that we are asking in terms of what we now know today as personalized medicine is we are all different, and our differences translate into the way in which we react to drugs as individuals. So the questions that we are asking is, why does somebody need twice the standard dose of a drug for efficacy? Why does a particular drug work for somebody but doesn't work for somebody else? Why does somebody get side effects other people don't? Why do some people get cancers but others don't? And why is it that anecdotal information or case reports that we read are maybe perhaps completely irrelevant to our own health and treatment? So what we are moving today, or have moved today in some manner, is personalized medicine. So with that, I'll talk to you over the next maybe 25 minutes or so about pharmacogenetics. I'll talk to you about pharmacogenomics and touch upon drug development. I'm extremely grateful to the Department of Pharmacology and Therapeutics at our institute for having given me this opportunity, as well as the organizers of PHARMASTEER and all of you who are listening in to this talk. So this is going to be the outline of my presentation. I'll begin by giving you a historical perspective, and then I'll talk to you about what is personalized medicine, give you an introduction. Then, just to break the monotony, we'll have a quick recap of fundamentals of genes, polymorphisms, chromosomes, etc. And again, to break the monotony, I have built in some quiz into this. I understand that I'm on a separate link, but we'll see what we can do. Why pharmacologists or clinical pharmacologists need to understand terminology in pharmacogenetics, some useful resources, and then I'll end with the summary of the presentation and what is the road ahead for pharmacogenetics. So if you look at the history or the beginnings of pharmacogenetics or pharmacogenomics, we know about Pythagoras, that's the triangle guy, an ancient Greek mathematician and philosopher. So Pythagoras actually talked about favism. He forbid the eating or touching of these fava beans, which we know causes favism, or this is associated with G6PD deficiency and acute hemolytic anemia, right? So this is really early, circa 570 before Christ. And then in the 1930s, 1900s, you had this chemist, Arthur Fox, who synthesized this phenylthiocarbamide. And when the dust of this phenylthiocarbamide actually flew into the air, some people had taste blindness. They couldn't taste things, right? So these were all the early beginnings showing that how one individual was actually different from the other. But it was in 1959 that Frederick Vogel actually coined the term pharmacogenetics after the discovery of the liver polymorphic enzymes. The early studies in pharmacogenetics, pharmacogenomics, as you call it, were done on the liver enzymes, the CYP family. And the one enzyme which has been maximally studied is the CYP2D6. Okay? The studies were conducted with drugs like debrisoquine or spartine. And in 1975, Smith and colleagues realized that there are differential responses in normal healthy volunteers when they use debrisoquine and spartine because of the polymorphism of this enzyme CYP2D6. It's not the most abundant, but it's the one that is maximally studied. And with the advent of technology, that is polymerase chain reaction, you had the speeding up of pharmacogenetics and greater appreciation of the understanding of the inter-individual variability in drug response. So if you look at what are the factors which finally determine how a particular person will react to a drug? It's the age, gender, the body weight, the race, the ethnicity the person belongs to. Is the person having concomitant drugs? Is there disease? Social factors, for example, malnutrition and genetics, right? So when you put all of this together, you have the science of pharmacogenetics or pharmacogenomics, the goal of which is basically to personalize medicine and make sure that you receive what is called as personalized medicine. So what exactly is this personalized medicine? It is the right drug in the right dose for the right indication, for the right patient, and at the right time, right? So I'm just going to quickly go through this quiz. I'll answer it, but let me just read it out. The potential outcomes of pharmacogenetic research include all of the following except. So your options are lower incidence of adverse effects, which is yes. New drug development, yes. Improved treatment outcomes, yes. And pre-treatment for screening for genetic polymorphisms. Higher healthcare cost is technology and therefore not an outcome of pharmacogenetic research. We then moved to 1953, where you had this discovery by Watson and Crick of the deoxyribonucleic acid. And if you look at the first sentence, which I've written in the title, this structure has novel features which are of considerable biological interest. And that sort of paved the way for the application of genetics in drug development as well as clinical practice. Let's now start with the DNA, right? So what is DNA? DNA is essentially a self-replicating material which is present almost in all living organisms and which carries your genetic code or your genetic information. And it has instructions which makes each species, including us, basically unique. And why is DNA important? Because it's inherited, but perhaps more important for us in terms of the topic today is it codes for proteins. And remember that a lot of human diseases are actually caused due to protein dysfunction. So let's look at DNA as the English language, right? I'm speaking to you in English. So let's look at it as something which represents the English language. Now, after this, what do you have in the language? We have alphabets, right? What are these alphabets? These alphabets are the nucleotides. So you have adenine, thymine, guanine, and cytosine, right? So the DNA made up of nucleosides, and these are your nitrogen bases, adenine, thymine, guanine, and cytosine. For example, a particular sequence might code for blue eyes, a particular sequence might code for brown eyes, and so forth. So these are your building blocks of life. What do you do with the alphabets? You put alphabets together and you form a word, right? What is your word? Your word is the codon, CGA, for example, as you can see on the slide, right? So the codon CAG, for example, represents the amino acid glutamine, right? A codon is essentially a triplet code which codes for a specific amino acid. Remember, I mentioned earlier that a lot of human diseases are due to dysfunction in proteins, right? So you have multiple codons, 64 codons, and 3 of these are your stop codons. After you have words in the English language, you need to put them together in a sentence. And what is your sentence? Your sentence is essentially the gene, right? So we started with alphabets, we've gone to words, and words are being put into a sentence, right? So what is your gene? A gene is basically the basic functional unit of heredity, right? And less than 1% of total genes are different between people. And these small differences contribute to me being me and you being you. So the unique physical features and genetic traits, of course, are determined by the 1% or less than 1% differences in the genes. And yet we fight amongst each other. Yeah, that's what we do, right? We go to war. Countries go to war, despite the fact that we are not really different. What is an allele? Something associated with the gene. It's essentially an alternative form of the gene, which is located at a specific position on a chromosome. And organisms have 2 alleles for each trait. Now, having looked at the sentence, we need to put the sentences into a chapter. What is this chapter? The chapter is essentially the chromosome. So this is a packaged and organized structure containing most of the DNA of a living organism. It's not found on its own, but a chromosome is generally packaged with proteins which are called as histones, right? So we've moved to the chromosome. And finally, if you put everything together, what you're looking at is the entire genome. All right. So we started off with English language where we had alphabets. That's the nucleotides. The alphabets became words, the codons. The words became sentences. That's the gene. The sentences were put together, packaged into a chromosome to make a chapter. And the entire chapter gives you your own life book, which is your own genome, right? And then the Human Genome Project, which ushered in the era of genomic medicine. So what is the purpose of pharmacogenetics or genomics and understanding all of this? Fundamentally, to improve drug efficacy, minimize toxicity. Prediction is one of its goals, of course. improve disease diagnosis and earlier detection of genetic predisposition to disease, right? All right. Now I have a quiz again here. I think I'll skip this. RNA contains the base adenine in place of guanine. The answer is false because RNA contains uracil. Identify the correct order of organization from the largest to the smallest. The largest is, of course, genome, right? So your answer is genome, chromosome, gene, and nucleotide. And I can't see the top. What can be described as a unit of heredity? Contains information to produce a particular protein, affects traits. Okay, the answer is all of the above. All right. Which is an alternate form of gene? The answer is the allele. All right. Now, the next thing that we come to is, if you remember, the title of my talk is pharmacogenomics, right? But I started off with pharmacogenetics. So there is one difference, fundamental difference, when we talk about pharmacogenetics versus pharmacogenomics. Pharmacogenetics is essentially a study of genetic differences in single gene, right? So when you're looking at how a single gene influences variability in drug response, either in terms of efficacy or toxicity, we're looking at pharmacogenetics. However, if you're looking at multiple genes or genetic differences in the entire genome, which affects or influences variability in drug response, either efficacy or toxicity, it's pharmacogenomics. So single gene, pharmacogenetics, more than one gene, multiple genes, entire genome, we're looking at pharmacogenomics. The next terminology I think we all should know is the distinction between what is a mutation and what is a polymorphism. A mutation is essentially a difference in the DNA code which occurs in less than 1% of the population, therefore associated with rare diseases, cystic fibrosis, sickle cell anemia, Huntington's disease, and all these rare or orphan diseases as we call them. But anything which is seen in more than 1% of the polymorphism, seen in more than 1% of the population, will become a polymorphism, and we shall see the impact of polymorphism subsequently. Then we have this term called as SNPs. Single nucleotide polymorphisms are essentially single base pair differences in the entire DNA sequence. And we are looking at over 2 million single nucleotide polymorphisms in the entire human genome. We'll now go on to the applications of this field of pharmacogenetics and genomics. And we look at these polymorphisms. What do polymorphisms impact? Just about everything. They impact metabolism, drug transport, receptor sensitivity, adverse drug reactions at safety, efficacy, responders, non-responders, and of course, disease susceptibility. Let's take one example of pharmacogenetics and drug metabolism, right? So if you look at the drug metabolizing enzymes, you have the phase 1 enzymes and you have the phase 2 enzymes, and you have this family, 3 families, CYP1, 2, and 3 of the cytochrome P450 enzyme system. And let's take an example of CYP2C90, right, which metabolizes approximately 15% of the drugs. And one of the drugs that this enzyme metabolizes is clopidogrel, which is a prodrug. Now, if you look at this New England Journal of Medicine article, now we're going to the application of pharmacogenetics. We are looking at cytochrome P450 polymorphisms and the response to clopidogrel. So you have a pharmacokinetic response and you have a pharmacodynamic response. So if you have mutant alleles of CYP2C9, the prodrug doesn't get converted to the active drug, and therefore you have reduced presence of the active ingredient, and you also have less blood thinning, as we call. So a lot of institutes, including KM Hospital where I work, have what are called as clopidogrel clinics, wherein you actually do a pre-prescription testing for these polymorphisms prior to initiating drug therapy with clopidogrel. Let's take another example of consequences of these polymorphisms, and that is safety, okay? That is adverse drug reactions. With the example of this particular drug which is used in HIV, which is called as abacavir. Abacavir is an NRTI, and approximately 5 to 8% of patients develop hypersensitivity to abacavir. And if you continue abacavir or you re-challenge these patients, then there can be fatalities, right? So there is a particular HLA type which is associated with abacavir hypersensitivity, right? That is 57 HLA B5701. And studies have shown that if you have patients who have this particular HLA type, then they are more susceptible to developing this hypersensitivity, which can be fatal. So one of the biggest impacts of pharmacogenetics is a very nice editorial which came in the New England Journal early on, I think 2010, that even if a fraction of the serious and severe adverse reactions can be prevented, the clinical and economic benefits of pharmacogenetics would be profound, right? So remember that pharmacogenetics, genomics is closely linked with technology. Technology is expensive, and therefore pharmacogenetics, genomics is also associated with pharmacoeconomics. So when you look at pharmacogenetics, you also need to see that if you're doing a pre-prescription genotyping, for example, to predict abacavir hypersensitivity, is it worth it? So this was a study which was done where they divided patients who underwent it. So you had patients who either tested positive for this particular 5701 or did not test positive. And then if you tested positive, you were given non-abacavir regimens. And if you tested negative, you got hypersensitivity or you did not get hypersensitivity. And they were able to show that pre-prescription pharmacogenetic testing is actually cost effective for preventing abacavir hypersensitivity, which is now today standard of care. And this is a, like a 2x2 chart that makes sure that when you test pharmacogenetics or genomics or using a particular test, if your outcome is good and your cost is low, that's when the test is actually maximally beneficial and you can actually bring it down to point of care testing. Difficulty is if you have a good outcome and the cost is high, and then we'll come to that a little bit later. Okay. Then we come to pharmacogenetics, genomics, and disease management, right? I'll give you the example of the use of trastuzumab for breast cancer. So this is a monoclonal antibody which is directed against the HER2/neu receptor, which is overexpressed in approximately 1/5 of the CA breast patients. Now, when the early studies were done in CA breast, actually, a heterogeneous group of patients were taken. And this was obviously before the discovery of this protein. Trastuzumab failed to show efficacy, and it was later found to be effective in a subset of patients, a subgroup of patients who actually had this overexpression of HER2/neu. So that is HER2/neu in terms of disease and in terms of targeting this particular protein with this monoclonal antibody trastuzumab. So if you look at this chart here, if you see the Kaplan-Meier curve of people who've been given 1 year of trastuzumab versus the standard of care, in those days it was nothing, you actually see a clear separation of the lines at 24 months in terms of survival, right? So that's how trastuzumab has got into pharmacogenetics. So you test for HER2/neu as part of pharmacogenetic testing or pharmacogenomic testing in patients with CA breast. So what is the application in terms of drug development? So what pharmacogenomics helps you to do is to identify new targets. It helps you to eliminate unsuitable targets at an early stage. It helps you to accelerate the process of drug development because you can actually enrich your clinical trials into a subgroup of patients whom you know are more likely to respond. And therefore, you have better study designs because you know where your drug or the product that you have is likely to work best. The next thing that I'd like to come to is, these terms are often used interchangeably. Is there a difference between precision medicine and is there a difference between personalized medicine? So a lot of what we do today, for example, if you have a group of CA breast patients, and you test them for this protein HER2/neu and you find positivity, you give trastuzumab. So what you're doing is you're tailoring your treatment towards a group of women. So when you tailor something to a group of individuals, that becomes precision medicine. Which group of patients is trastuzumab most likely to work in? So this is obviously based on genetic, environmental, and lifestyle factors. Whereas when you do personalized medicine, what you're doing is actually personalizing it for that individual, which is the ultimate goal of pharmacogenetics. And here on my left, I can see a lot of shirts, right? So you have collar sizes in men for 38 or 40 or 42 or 44, right? So when you go to Shoppers Stop or West Side, what you're actually practicing is precision medicine, right? Because you go into a counter, you find your shirt size, and that shirt size fits a group of individuals. Whereas if you go to the tailor and get your shirt tailored, what you're doing is personalized medicine. With all of this, you might actually wonder, there's so much to learn in pharmacogenetics, so much to learn in pharmacogenomics. So is it mission impossible? A lot of it was not there when we were studying. A lot of it has come in the last maybe a decade or more. So I think as students of pharmacology, clinical pharmacology, it's good for you to know resources which are available for you to learn. It's good for you to have some understanding of terminology. And it's good for you to know a little bit about gene therapy because there is so much of explosion of gene therapy in the market. So what are the resources that all of you as students can use? The first, and I think a very fantastic resource, is the US FDA website. So if you go to the US FDA website, there is a very nice section on pharmacogenomic biomarkers in drug labeling. So if you look at this abacavir, which I mentioned earlier, they talk about the therapeutic area, which is infectious diseases. The biomarker is HLA-B. The subgroup is HLA-B 5701 carriers. And the the outcome is the change in the product label. So there is a boxed warning, there is a contraindication, there are precautions. And if you look at the product insert, what it actually tells you is that you need to test for HLA-B 5701 prior to prescribing abacavir. So this is your US FDA website. Another very, very good website, which something that I refer to pretty often, is the CPIC website. Yeah? This is the Clinical Pharmacogenetics Implementation Consortium. What is this CPIC? It is essentially it's free, it's available in the public domain, international consortium of individual volunteers. There are a small group of people who actually curate, create, and post free evidence base on pharmacogenetics. Okay, since we have a therapeutic drug monitoring clinic in our institute, I've taken the example of phenytoin from CPIC. So you can actually search the website for individual drugs and pharmacogenetic information about these drugs. For example, if you look at information that is available in CPIC, it tells you what happens if you're an intermediate metabolizer of So phenytoin is metabolized largely by CYP2C9, and partially by 2C19. So for intermediate metabolizers, it says use the initial dose, loading dose, but subsequent doses use approximately 25% less. If you're a poor metabolizer, use the typical loading dose and subsequently use approximately 50% less. So the CPIC is very, very useful if you're a practicing clinician and you think a particular drug is susceptible to this. So a particular individual has susceptibility, right? Okay, the next database, which is important, is a very nice database, Pharmacogenomics Knowledge Base. So it is GKB, it's not KGB, right? So Pharm GKB, again, a very, very useful website for pharmacogenetic information. These are just examples here, but there is a lot of wealth of information available, but a lot of these websites are commonly used by clinicians and researchers to look up pharmacogenetic information. The next thing I think as individuals you need to know is that gene therapy, right? I think the first gene therapy approval was in China, way before Europe and US. But this is an example of this tisagenlecleucel, which is FDA approved. And this is FDA approval for the first gene therapy in the United States. So as students of pharmacology going forward, you will see a lot of gene therapy products. So you need to have some understanding of what this is all about. Therefore, you need to have a bit of reading around it, right? So this is actually shows you this slide, how gene therapy and gene therapy products have evolved over the years. 60 years was spent in basic research. And then I remember when we had finished our MD pharmacology, there was this huge hue and cry about the death of a patient in a clinical trial who actually got multi-organ failure. It's there up on that. So, then it went down a bit, and again, it's flourished now. So if you look at the gene therapy products which have been approved in the last 20 years, there are so many of them. And you are the future of this country in the next 10, 20, 40, whatever number of years. You will see a lot more, tens of thousands of new, not thousands, but lots of new products are going to come into the market. So you need to keep abreast of gene therapy and the way it is going to change the way we practice medicine. There has been criticism that India doesn't do as many gene therapy trials as, say, elsewhere in the world. But I think that landscape is also changing now. If you look at the CTRI, the Clinical Trials Registry of India, and this is a June 2021 article about the first supported CAR T cell therapy, which is being conducted in ACTREC in Tata Memorial Hospital. And I'm sure that we'll see a lot more gene therapy, a lot of studies going on all over India, right? And I'm sure we'll see a lot more of gene therapy, including products which are developed within India, manufactured within India, trials being supported by government and other agencies in the country. So now, if you looked at the 1900s, you had the germ theory, then you had chemistry, you had physiology, pathology, and physics. So your whole thing was about understanding disease. You find it and you fix it, right? Whereas in 2000, you have genomics, proteomics, metabolomics, something which is not written, transcriptomics, systems biology, spatial biology, bioinformatics, micro nano processing, and I don't know what other terms. Yeah. So you're not just understanding disease today. What you're actually doing is you're understanding health, right? And the complexity of staying healthy. So what you're doing today is predicting something, and you're also personalizing it. So you can see the change between the 1900s and 2000. So what happens to you all, right? I mean, all of you who are listening. This was a statement made by Sir Francis Collins in 2004, and I think it's still true. The explosion of information about genetics is going to create a huge problem in health education. Most physicians, and I would even add pharmacologists, have not had a single hour of education in genetics, and therefore are going to be severely challenged to pick up this new technology and run with it. Yeah? And as pharmacologists, I think the statement holds true for us as well. We need to keep abreast of it. So whenever I take talks on statistics, the question that is asked to me is, why should I know about it? I don't think it is why should I know, it is how much should I know about it. And I think the answer is the same for pharmacogenomics. So what I would recommend all of you to do is have a little bit, not too much. What does CRISPR mean actually? What does Sanger sequencing mean? What does next-gen sequencing mean? I'm not saying go into it entirely, but have some understanding. And what I've not discussed, and which is an area by itself, is use of artificial intelligence in pharmacogenomics, right? What is artificial intelligence? For example, in cancers, what they do is they look at the microenvironment in tumor samples to see if they can actually tailor drugs which are going to impact the tumor. So there is going to be a significant explosion of artificial intelligence, like in many other areas in pharmacogenomics as well. And therefore, to sort of end with Charles Darwin, it is not the strongest of species that survives, right? Otherwise, the dinosaurs would have lived. But the species that survives is really the one that is most responsive to change. And with that, I'd like to thank the people from whom I have learned, and special thanks to Adityan sir, under whom I learned pharmacogenetics and pharmacogenomics for the first time. I would like to thank the organizers once again, and I'm hopefully on time to take a few questions. Thank you, everybody.
Thank you so much, Dr. Nitya, man, first of all, for being here, and then for sharing your critical insights around pharmacogenomics, which I'm sure that is going to reshape entire healthcare ecosystem in days to come. Ma'am, we do have received some questions from the students while your session was going on, and my first question to you is, what emerging technologies or methodologies are showing promise in advancing pharmacogenomic research for new drug development?
So actually, I did speak about some of the technologies. One of the most important technologies, I think, is the sequencing, which is now available. I remember when we started our careers, something like the polymerase chain reaction was really huge for us. And we started with a methodology called as RFLP. Now we have this whole genome sequence. And sequencing, I talked about Sango sequencing. The other thing, which is, I think, coming up, one of the things I did not mention in my presentation is that we must not forget that diseases are not monogenic, as in it's not just one gene leading to disease. We're looking at polygenic, so when something is polygenic, then it becomes a little more difficult. So the CRISPR technology, which I talked about, that is gene editing, is going to be applied now for drug development in, these rare and orphan diseases like Huntington's disease or sickle cell disease and so forth. So technology like CRISPR, for example, is something you keep reading, no? Cas9, CRISPR, Cas9. So there is going to be, and of course, you have bioinformatics with so much of information. How do you process this information? Proteins are upregulated, proteins are downregulated. I think there is a lot. I'm just giving you a few examples. I'm in no way an expert on all of this. Like I said, therefore, our understanding of at least what these technologies do will become important for us going forward. Yeah.
Thank you so much, ma'am. My second question to you is, how can pharmacogenomics be effectively integrated into existing healthcare systems to ensure widespread accessibility and the benefit?
I think anytime in India the question about access is raised, it is linked with the cost. Yeah, anytime, anything you say, oh, we don't have access, it's about cost. I think it's about making technology available. I represent a public sector institute. I'm sure in private sector, a lot of this must be already available. You have to bring down the cost of technology so that you can bring it, closer and closer to the patient. So in our institute, for example, we are able to deliver pharmacogenetic services for CYP to C9 polymorphisms or to C19 polymorphisms or for warfarin for the VQRC. What saddens me, unfortunately, is that it's almost always post facto. So if somebody has bled with warfarin, they come to us and we do the pharmacogenetic testing and we find that they had a mutant allele. But if we were able to do the pre-prescription testing, we could have actually avoided it. So sensitization, I think it's also important that we educate our physicians that these facilities are now available. So please use them rather than going by trial and error. And ultimately, technology, we need skills also. We need skilled people to do these days. So I think it's a mixture of a lot of things, technology, cost effectiveness, all of which education will enable access finally for patients. At the end of the day, we must not forget that the person who is impacted is that one person, right? Yeah, you may say that 99 people don't get an adverse reaction. For that person, his adverse reaction is most important.
Right. Thank you so much, ma'am. And my last question to you is, are there any challenges or opportunities associated with implementing pharmacogenomics approaches in diverse ethnic or geographic groups?
So there are studies now which are done in many countries. For example, a lot of work on pharmacogenomics is come from South India, pharmacogenetics and pharmacogenomics, which tell you about the variation, between South Indians or Maharashtrians or Gujaratis. So similarly, we have information from elsewhere in the world. Country-specific polymorphism information is available. So the ICH, I think it's the E5 guideline, efficacy E5 guideline, which talks about ethnic differences in translating drug development from one population to another. So there are guidances available on this E5R1, I think is the guidance. So we need to make sure that what we do is also in line with the guidances which are now easily available.
Thank you so much, ma'am. With that, let me thank the students for asking great questions. And thank you so much, Dr. Nithya, ma'am, for being here with us and patiently responding to all the questions that are coming across. Thank you so much, ma'am. With that, let me now move on to our second topic of the day, and which is market access and health economics. And for that, we have with us Dr. Ananya Biswas, Manager Health System, AstraZeneca India. Dr. Ananya is a dedicated professional with a wealth of experience in health system solutions. As a seasoned expert, Dr. Ananya is leading the design and implementation of innovative solutions aimed at dismantling access barriers for the patients in both public and private health sectors across India. Dr. Ananya has also done Masters of Public Health from TIS Mumbai. And with 8 years of experience, he has also excelled in the roles at World Health Organization, demonstrating her prowess in health economics and outcomes research. Ananya's impactful journey underscores her commitment to driving positive change in healthcare access. With that, let me hand over the forum to you, Dr. Ananya, for steering this session forward. Over to you.
Thank you so much, Ashish, and thank you so much, Dr. Nitya ma'am, for setting the stage. I mean, the last question that was shot at you kind of build the stage for access. I apologize a bit for the background noise. So, Ashish, can you just confirm if I'm audible clearly and
Yes,
my
you are.
voice is loud?
You are.
Okay,
You are.
great.
It's perfect.
So, I'll share my screen, and before we go into the presentation, just a second. Also, let me know if my presentation is visible.
Yes, Dr. Ananya, it's visible.
So again, it's a very simple presentation compared to what we just saw, data loaded and information loaded. Because one of the reasons for it is because the information and the domain is very niche. So if you talk about India in particular, and even the world in general, market access is a very growing and it's a very new domain. So what does market access mean? Now it's very difficult to put a definition to it. Like if you would want me to put like a short paragraph of definition, is very difficult, but I would tell you the end objective is always to make like the drugs, the newer technologies available to our patients. And as ma'am said, one of the major barriers is always affordability, but that is not only the only barrier. So affordability, yes. Accessibility, as in even if a person can afford, is the technology available in India, or does that person have to go to US to kind of get that medicine, to get that test done? And acceptability. So some newer technologies, some newer medicines might still not be acceptable. Despite being very effective, studied very effectively. And how should the communication be designed around it so that they can become more accessible and more acceptable to the population? So I'll tell you about the AstraZeneca vision in market access, and that is to lead by shaping the payer environment. And I will go into details and as in what that payer environment means, to create and deliver highly valued market access solutions. Now what does that mean? I mean, this sounds like a lot of words, but what does actually it means? So if you go back a bit to Dr. Nitya ma'am's slide, you will remember she spoke about precision medicine and personalized medicine, right? But again, that is just the technology part. How do you make any solution, any newer treatment more accessible, more affordable by everyone? And we talk about individuals who might be in the end stage cancer, or I'm particularly looking at rare disease. So rare disease, how do you even help the journey of diagnosis become easier for patients? Because we understand that the prognosis depends a lot on which state the patient was diagnosed. So this kind of highly valued market access solution, so that solution should be valued and should be acceptable to the market you are catering to, so that we can unlock the potential of our life-changing medicines for patients. So that is the basic idea of any market access role, the basic vision. Different corporates, different drug houses, different pharmas use different terms. They can be market access, they can be patient access, but they all evolve around the same idea, that we have to make our drugs, our technologies, including diagnostics, more accessible to the patients who finally need it. So I'll move on to the next slide. This is from the NHA estimates, 2000, and this is a collaborative report. You can see I am looking at almost a decade, a bit less than a decade of data. So 2013 to 2020 data. So this is how the pair evolution has happened. Now this is very important to understand. We always talk about, India spends so much less of its GDP. So we always talk about India spending 3% of its GDP or less than that in healthcare. But again, that is the government spending. What is the out of pocket expenditure? Does that amount to a number which is more viable? So yes, it is. The out of pocket expenditure in India is high, and I'm sure all of you being doctors, as well as having gone through the journey yourself, sometimes might have realized that we have to spend out of pocket. So how do we kind of reduce it? How do we kind of make those solutions more easier for the population of India, so that they can access newer technologies and newer medicines and newer drugs without having to pay exorbitant amounts from their pockets? And how can they not be pushed into poverty just because of a health crisis? So as you can see, just a second, let me put my pointer on. Yeah. So as you can see, this line, it kind of shows the out-of-pocket expenditure. I don't know how to move this. Yeah, out-of-pocket expenditure, and this blue graph line, it shows the steady increase in the government health expenditure. So this is a good scenario. I mean, this kind of a graph is something which gives you hope. An ideal situation would be when the blue line goes higher up and kind of crosses the orange line altogether. But at the same point of time, this is very optimistic a graph, and it shows that the government health expenditure is uh, increasing. And that also means, that also translate that the government is looking at newer avenues of spending. So the government is looking at including newer molecules into their formularies. The government is looking at having to cater to more diseases. So if you have heard about the national policy for rare disease, you would know that patients are getting 50 lakhs for their treatment, and the government is actually trying and pitching for that amount to go higher. Like the ministry is trying and pitching for the amount to go higher. What is a bit grayish in the slide is the gray line that we see. Private health insurance. The risk pooling attitude of Indians is still not something that we should appreciate. So health insurance, and when you talk about disease specific health insurance, so suppose there are companies which are now catering to cancer specific insurance, or mostly cancer specific insurance. So that uptake is dismally low, and obviously that also acts as a barrier to access newer technologies and newer medicines. Hopefully this risk pooling attitude with further education and further spending capacity, and not only spending capacity, the education that goes behind spending capacity would increase this graph. But as of now, you can see over a decade the growth is almost negligible. Any questions from this slide? I mean, I would like to have it as a more interactive session. So if anyone wants to understand what this data is, or how we are extrapolating or reading this data, I'll be more than happy to answer. Ashish, you have to help me with the fact if anyone is raising hand, because I am not able to see the screen.
I'll do that. Meanwhile, I think we can proceed if any questions would.
But please feel free to interrupt me. I want this to be more of an interactive session. I mean, this should be more like a peer talk. We are here to discuss. So again, this is another graph that will probably excite you. So we'll go rings by rings. This is the 2017 data, like 2017 to 2018 data. The next concentric ring where my laser pointer is now revolving is the 1819 data, and the last one is 1920 data. Now, if you look carefully at the concentric rings, you will see that the orange part of the ring is slowly increasing, which means state government health expenditure is increasing. Now, at the end of the day, health is defined as a state subject. So when state governments decide to put a greater amount of their expenditure into health, it means easier accessible solutions which are more tailored to the population of that state. We have to acknowledge here that, say, for example, the health needs of Kerala would not be the same as the health needs of Manipur or Mizoram. So if states are spending more, and obviously union government is spending at par, but if the percentage share of health expenditure from state governments go higher, that means the governments are interested in investing in health infrastructure. And this also leads to a very cascading effect. Like, let me tell you for an example. So for AstraZeneca, right, we have collaborated with like government of Kerala for a center of excellence, and that became kind of a role model. That became kind of an example which other states wanted to emulate. So they also wanted us to kind of join hands with them and sponsor a bit of not a bit, actually sponsor the entire AI generated imaging technologies. So if you have heard of Cure AI, this is a very recent and very promising platform where like AI algorithms or artificial intelligence algorithms are used to just look at scans and kind of scan a small nodule, so which leads to diagnosis of lung cancer in very early stages, before metastasis happens and before the prognosis gets worse. In those kind of states, if the diagnosis is done early and the treatment begins early, the overall survival is much, much higher in patients compared to a late stage diagnosis. And that is obviously general knowledge. But with state governments kind of putting that money in, emulating this kind of models, taking up this kind of newer AI generated technologies, always at the end of the day, it always helps the patient, the public, it always helps us, the citizens of India. I'll move on to the next slide. Just a second, I'll just take a sip of water. Any questions so far? Nitya ma'am, would you want to add something or say something? Okay,
Yeah.
I'll move on. Yeah. Again, these are some market access key focus areas, very broad umbrellas, and is beyond the scope of this meeting to kind of go in depth into all of them. But what I'll try to do is I'll try to exemplify so that you can at least relate to something. So first is increased reimbursement access. Okay, let's take the example of rare disease. So if you know about all the government policies that are ongoing right now, so Rashtriya Arogya Nidhi, Pradhan Mantri Yojanas, and even the latest, the PMJOY, so Jan Arogya Yojana. So they all have a capping limit. It can be per individual, it can be per family, depending on the state or depending on how you're enrolled into the program, but they all have a upper capping limit. But when you look at the national policy for rare disease, having acknowledged that rare disease is a much, much costlier to treat and difficult to treat domain. First of all, you don't have the medicines. You have very few rare diseases which are actually curable through medicines. Most of them are just palliative care or kind of supportive care. So in those cases, the cost of treatment is very high compared to any other disease. So what the government did, they increased the value from 10 lakhs to 50 lakhs for treatment. Now, is 50 lakhs enough? Yes, in some cases it is, but in some cases it not. But it is at least the 5 times of what the government is paying for other diseases. So, and how did that happen? Did that happen suddenly? Did the government suddenly decide to do it for one particular disease area? No. It took a lot of advocacy. It took a lot of patient advocacy groups, pharma companies coming together, voicing their concerns, pharma companies actually coming up with newer methods so that they can decrease the price for Indian market, and at the same point of time both coming somewhere and meeting midway so that patients get benefit at the end of the day. Self-pay access expansion. Now self-pay access, so self-pay is basically out-of-pocket expenditure. So as a payer, as a patient, I am the one who is paying for it. There is no reimbursement coming for government, or there is no insurance reimbursement that is coming as well. So for a self-pay patient, obviously the burden of disease is also a huge part of the burden of disease is also the economical burden of disease. People lose wages, they kind of have to drop out of the workforce, so the earnings decrease. At the end of, and at the same point of time, they have to spend so much more on their health, right? So we have seen a lot of studies, and you can go to the World Bank site, and you can see that how many people actually drop down below the poverty line just because they had a catastrophic expenditure for health. Now it is something of concern, so there needs to be addressed. So again, giving an example for the same, and again, this is not the holistic view. There are so many ways we can do it. There are so many stakeholders involved, but one of the ways that pharma companies generally do is they provide patient access programs or patient affordability programs. They're generally called PAPs as an acronym. So patient affordability programs is the patient affordability programs is basically when pharma companies decide to give a free vial or a free strip of medicine on the purchase of one. But this is very catered to people who are just paying out of pocket and not for reimbursement sectors. Third, support launches and build innovative value solutions. Support launches. So and in the next slide, we'll go more into what launches mean and how a majority of our work happens before the launch of the medicine. But in a very general way, launch has to be something that kind of comes with a big bang, and not in like a real sense of the term, but educating your prescribers. They need to know, our doctors need to know what to prescribe. So that is a major part, and that is the reason all pharma companies have a very committed and very talented team of medical affairs, because that is where they all go out, all trying to reach as many healthcare practitioners as possible and educate them. And innovative value solutions, again, innovative value solutions is again very domain specific. Will it be same for the vaccine as for an oncology drug? No. But is there some general example I can give you? Okay. Say for vaccines, so one of the vaccines that we are planning to launch, we are trying to look at the states which have a higher burden of pneumonia and higher deaths because of pneumonia and poorer health infrastructure. So let's take an example of Bihar and Kerala. So Bihar, the burden of pneumonia is high, right? and poorer health infrastructure, very fewer tertiary care centers. And we also what happened in COVID in Bihar and UP. At the same point of Kerala, so lower burden of disease, but better health infrastructure. So would a government be which is struggling with tertiary healthcare sector and doesn't have enough resources to allocate to it, along with the double burden of disease, be interested in having a solution that is more catered towards prevention? Yes. So again, innovative value solutions have to be very precisely catered to the molecule, to the drug that we are launching. And beyond the pill solution. So beyond the pill solution varies a lot, but two of my favorite examples and something that we recently launched, and Ashish, we are going to talk about it in details in our national strategy meet. But just to give you guys here a brief, so we have a molecule, you have a drug, and it requires, say, some sort of hormonal testing or some sort of genetic testing. Sorry, excuse me. Very costly and very precise. Maybe one medical college in the entire state is doing it. Sometimes not even that. Maybe just the metro cities are doing it. Can we have tie up with governments, help in the reverse cold chain process, and ensure that samples from very remote areas and remote districts reach those metros, reach those few centers in India? Yes, we can. And that is one of the strategies here. Can we ensure that we have dedicated trained workforce in these remote areas who can at least refer properly or collect the sample properly and pack it so that they follow the cold chain system? Yes, we can. So these are one of things. Secondly, we all know that oncology, any cancer diagnosis is not easy, neither for the individual nor for the family. So we have programs where we provide nutritional, psychological, as well as a kind of a navigator support to the patient. So the person will have telecounselors, and the person will have people who will, dietitians will give them very personalized catered diet for their oncological treatment or whatever nausea, vomiting, and side effects, adverse effects that come with an oncology treatment. They will have psychosocial support. Sometimes it extended to the caregiver also. In certain cases, if it is a pediatric indication, we extend the psychological support or the counseling support to the caregivers as well, because it's not easy for any parent to see their child go through something so terrible. As well as a patient navigator who will help them navigate. So, those questions, okay, where do I submit this bill? Which clause in the insurance to be kind of put up? So this kind of knowledge, navigation knowledge that patients need. I'll move on to the next slide. I'm hoping no questions on this one. Okay. Now this is a more detailed slide. And again, this is nothing very new, but this is the strongest pillars of market access and pricing function. So first is access strategy. Access strategy has to be built from the very beginning of a launch, like much before a launch. So it generally starts, varies from molecule to molecule and domain to domain, but generally starts at least 3 to 5 years before a launch. It obviously starts 3 years before a launch. And what do we do there? We will look at this. So I want you to move to my early asset access strategy. This first of all, an environment shaping. Do we know who are the prescribers? Are the patients going to the right prescribers? Is there any other medication which is not indicated and which is not approved, which is off-label, but being used without its, efficacy and safety being studied? So this kind of questions need to be answered, because if there are this kind of challenges, they need to be addressed. People need to be educated. Epidemiology. Epidemiology is like one of the key pillars. So that will not be only clinical epidemiology. We're not only looking at incidence and prevalence. We are also looking at regional variations. Sometimes we look at age variations, and all that come as a very core part of epidemiology. But regional variations is something we look at very strongly. Also, is there any difference between a certain group? So are people who eating some kind of a different cuisine, are they at a higher risk of cancer? And what is the way we can reach those people? Because if they're at a higher risk, so they need those medications more, right? If they're having higher prevalence or higher incidence. So that kind. Unmet need and burden of disease, now huge. Sometimes it is a very difficult domain to study, because unmet need, one of the biggest, and I keep on saying this, one of the biggest unmet need is because we don't have enough tools or enough resources to study it. So that has always been a literature gap. And for newer diseases, for evolving diseases, for rare disease, newer cancers, more difficult to treat cancers. That has always been a big gap and a big question. Pre-launch planning has actually involves a lot of again stakeholder mapping, stakeholder engagement. So that is what is co-create business cases. Business cases means that okay, if I have to pitch to my global body that I need to give 1+1 free in India, because India's population's affordability is here, and the drug is priced at such that India cannot afford, or the patients in India cannot afford the pricing at that point. So I have to create a business case saying that we have to give one free and we have to get it validated. Dossiers and health economic models. Now, again, dossiers in India is not yet a very widely used concept. But if you, and I mean, thank you, Ashish, you mentioned WHO, but I began my career with Novartis. I worked for Novartis and then I went to WHO and now AstraZeneca. So in Novartis, when I worked there, I used to like prepare documents for FDA and EMA. So when you look at those documents, those dossiers, they have to be very detailed. But if you ask me, what is the scenario in India right now? I would say it's very nascent. It's very nascent. It's just evolving. The government has started to realize the need, and Dr. Soumya Swaminathan, who was in ICMR, and she had kind of built this center out of Vizag, in Andhra Pradesh, where she wanted to look at health economics and outcomes research and health technology assessment. But then again, she moved to WHO. So it's evolving. It's in the very nascent phase, but that also tells you that it has immense potential. So if you are building the skill sets, they have immense potential in the future. And again, business access strategies, again, innovative value solutions, which I told you is catered. So access strategy, pricing. Again, pricing bit is very catered. There are certain global guidelines that we have to follow while pricing a drug. It cannot be just out of the whims of a person. A drug pricing has to go through multiple layer of approvals, both internally and externally. So it cannot be just and pricing strategy that happens. It has to get validated by governments, by reimbursement bodies. In India, we follow the NPPA, and I'm sure all of you are aware of it. So the NPPA is the one which guides the India pricing policy. And if you ask me one major difference about global pricing and India pricing, global pricing allows you to have differential pricing for one molecule. So suppose I have one molecule, let's say molecule X, and it can treat ankylosing spondylitis as well as it can treat psoriatic arthritis. You are allowed to have differential pricing for this differential, for these different diseases that it caters to, depending on the burden of disease that you have already established with the payer. But in India, you are not allowed to do that. In India, we follow one price for one molecule, and it can cater to a number of diseases. It can be a panacea for many, very many diseases, but it will still have one price point. So yes, PSP, PAP, I spoke a bit. So PSP is patient support programs. PAP is patient affordability or patient access programs. They are both used interchangeably as a term. Patient support programs is basically any program which is not involving the price, or as if not involving this 1+1, 1+2, 2+1 kind of a strategy. That is PSP. So examples of PSP, you're helping in diagnostics. You're setting up diagnostics facilities. You can't set up maybe, but you are tying up with vendors. You're making it more accessible. You are creating educational material. You are creating flyers. You're leaving behind leaflets at your healthcare practitioners so that they can give those coupons to their patients and help them get a free drug, free testing done. For example, take this as an example, okay? So if it is a rare disease, okay, and that disease needs genetic testing. So this is a Sanofi example. Sanofi was working in SMA. So that needed genetic testing. So what they did is they realized there's a huge gap. There is not enough geneticists in India. And as ma'am just spoke about, very niche domain. We are growing, and we are growing at a very good pace, but we are still growing. So we have not reached there. So there are not very many experts in India who can actually work on it. So what Sanofi did, they tied up with the Center of Health and Genetics in Bangalore, and they started a postgraduate diploma. So for all MD patient, for all MD students who have passed, successfully completed their MD examination, they can apply for the diploma and they can become a genetics, like a specialist in pharmacogenetics. Genetics. So again, that is one of the PSP examples, and I'm going a bit beyond my brand, and I hope Ashish that is okay, because this is a very educational. So these are kind of PSPs, and then there are say the kind of program I just spoke about is called Sahai, which means support. So comes very strongly from patient support program. In nutritional support, psychological support. In one of the molecules we have, the indication is pediatric, and because it's pediatric, so it's very difficult to talk to children, right? They don't communicate emotions the way adults do, and even, yes, sometimes adults also have difficulty opening up, but they at least know how to communicate. So children don't know how to kind of palpate those emotions and communicate. So what is the easiest way to calm their anxiety, calm their stress, calm their the burden that they're feeling about being diseased? The best what we figured out was sound and body sound mind therapy, or sound body therapy as you call it. So some kind of, we see in Buddhist the sounds and the chanting. So those kind of sounds policy is something we looked at. Health financing, again, depends a lot on the payer structure. The health financing policies that are good in Spain are absolutely useless in India. So accordingly, this has to be very market specific, specifically catered. One of the best ways to learn about is to go through some of the reports that KPMG keeps on publishing about the payer evolution in India or in any country, and understanding how different countries pay for their health, and how it actually affects the outcomes, how it actually affects as a patient, how can I get a medicine, or how can I get a test done, and how easily I can get it done. So health financing structures. Public health initiatives is very largely about building the health infrastructure. So let's say pheno testing. Okay, so pheno testing is for severe, is for asthma, so some of our indications. So not available even in some of the top-notch facilities or top-notch corporate hospitals in India. Can we do that? Can we help it set up? And can we sustainably fund it so that patients have access, even if it's like, say, 4 or 5 metros across India, because it's non-existent, right? So something is better than nothing. Can we do it? And those kind of public health initiatives. So can we have, say, diabetic educators? Because patients with diabetes are most often not very well educated in how to manage it. They sometimes are obviously non-adherent to the medication, but even if they are very adherent to the medication, their diet, their lifestyles, their exercising, all of this varies. So can we have diabetic educators in government facilities? Because doctors are busy, they are overloaded, the outdoors are overflowing, they're crowded, right? Can we have a kind of a extra human resource who will be specialized in educating about these NCTs? So these are some of the public health initiatives. HUR, one of my favorite and most interesting domains, involves a lot of reading, a lot of research. But health economics and outcomes research is basically, put very shortly, how you define what you spend versus how you define what you get. So if either as a patient or as a government or as a corporate hospital, I am spending X amount of money, what is the quantifiable amount of health benefit I'm getting? And there are different ways of quantifying the health benefit. There are QALYs, there are DALYs, and you all are very well versed with it. So I'll not spend time on it. But you have to, and again, these studies and these models have a lot of assumptions that go into it. So taking all those assumptions, validating all those assumptions, ensuring that your model is as robust as it can be. Health economics and outcome research is basically one of the core pillars into how your global value dossiers or your value value proposals, or say your value stories. Value stories are the most commonly used tool in India. So value proposals and value stories go out to the person who is paying for it. So yeah, we've almost done this slide. I'll just quickly go through a bit of the part. So pricing, I told you, pricing starts from a very early stage. PAP price structure, cost of therapy. If there is an early access program that is needed, so early access program is again before the drug is already launched in India, or say in any country, in any market. Can we get it in as an early access? Importation program prices, how they're different, and bridging programs. So suppose we have patients in Kenya and they don't have that medicine, but that is available in Zimbabwe. Is there a bridging program that we can do so that patients can actually get those medicines and at a very, very lesser cost, like they don't have to pay much taxes to the government on it. Price operations are done. Sometimes they are voluntary price revisions, sometimes they are mandatory price revisions, and mandatory pricing revisions are done by NPPA. So NPPA time to time kind of mandates a list of molecules that need to go through some price revisions because they have biologics, so biosimilars in the market, or they have more generics in the market. So the molecules have to go through price revisions. And as I already told you about flat pricing, India follows a flat pricing structure. In a market brand access solution, so this section happens once the brand is in the market already. So again, there are awareness. There can be campaign, there can be public health awareness, campaign targeted to doctors, to prescribers, campaigns targeted to patient access groups, campaigns targeted to patients. So they can, depending on the need, the campaigns vary. Diagnosis programs and health support systems, public health again, treatment, PAP, financing, insurance. Insurance again, as I told you, is in a very dismal state in India right now. So, but can we increase the education around insurance? Can we teach people about risk pooling and reimbursement, commercial discounting, that all comes into it? And all of these programs have been monitored very strongly. They're not only monitored by the pharma company that initiates it, they also undergo regular, thorough audits and real world evidence studies. So once the brand is in the market, right? We have completed phase 1, phase 2, phase 3, and phase 4 is also going on when the drug is in market. But at the same point of time, there needs to be some real world evidence studies, and again, huge umbrella, but mostly focused on treatment adherence practices, prescribing practices of how patients report their outcomes, and all of this. I'll move on to the next slide. These are the major tools for HEOR. As I said, HEOR is one of the major key pillars in how all of this is communicated. There might be a zillion things, but the way you communicate it to the payer, say to the FDA, to the EMA, to the Ministry of Health and Family Welfare, depends who your payer is, but the way you communicate to the payer is of critical value. So these are some commonly used tools. There is systemic literature review, there is targeted literature review. Generally, global value dossiers contains systemic literature review if it's going for a approval, but sometimes it's just for a revision or addition of a newer indication. It can go for a targeted literature review as well. Value proposition, value stories, and network meta-analysis. I'll just briefly explain about network meta-analysis, assuming that you are aware. So suppose there is a clinical trial which compares drug A to drug B, and there is another clinical trial which compares drug B to drug C. But you need to, your payer, your government, or your insurance provider wants to look at drug A versus drug C. Now, there is no direct study, because there is a direct study between drug A and B, and there is a direct study between drug B and C. So how do you create that? That methodology is called network meta-analysis. It's a bit complicated. You have to standardize all the outcomes. You have to standardize all the sample population. You have to go through methods of standardization of sample sizing and everything. But that is how network meta-analysis is done. And health economics, so it contains this economic models part. Again, they're divided into some major segments. Just a second. Some major segments. There's a budget impact model. What is budget impact? Simply said, I'll pay X rupees. How much will it get me? What are the number of patients I can save if I spend 1 crore rupees? In budget impact model, there is generally not no competitor involved. Suppose I am a drug X, and if I spend some million rupees on drug X, how many patients we can save? What is a cost-effective model? Cost-effective model generally involves a comparator. There is always a competitive drug that is involved. So suppose there are two drugs, X and Y, and if I spend 1 crore rupees again on drug X, will it be more effective? Or if I spend 1 crore on drug Y, will it be more effective? So these are cost-effective models. Cost-efficient models are almost on the similar lines, but they also look at a bit beyond the clinical outcomes. So they also look at sometimes they look at patient-reported outcomes and all. And quali and DALI studies, which are the most primary basic studies, basically, and sometimes they're a part of the model. So quali and DALI studies are basically a stepping stone so that we can move ahead for the budget impact or the cost-effective or any of the CE models that we go for. I'll move on to my next slide. Skill sets. And when I was just actually listening to Dr. Nitya ma'am, I realized that I missed out one point because her slides captured it so well. Her slides actually captured it so well. So I will talk about it. And that is the first point, ability to interpret, analyze, and extrapolate data. So not only to interpret, analyze, and extrapolate data, you also have to have the capability of triangulation. Uh, Data triangulation is a very important skill set, and we often as students miss out. So if you're looking, say suppose if you're looking at most commonly available data set, maternal and child health data sets, make sure you look at all the published studies, government databases, and now they have launched HMIS, HMIS. Now they're trying to integrate it into IHIP platform, which is the Integrated Health Information Platform. And again, that is a baby of WHO in India. So they're trying to make it more integrated as a platform, not only limited to IDSP. I'm sorry, I'm using a lot of acronyms, but I'm assuming since all are here from the same domain, we all are very well versed with these acronyms. So this kind of data, I have come across studies where they look at a single center data, or even if an entire district study, but they do not look at the HMIS data of the district. And thus the study fails to go through peer review because you missed out a major point. So this ability to interpret, analyze, and extrapolate data is one of the key skill sets that you need to build. Again, while building this, you need to read through a way too many research articles. And and I have always followed this in my life. When you feel that, okay, I have started knowing a little about this subject, you start reading in depth. And by the end of it, maybe by a month of reading in depth about this subject, you are most likely to feel that, okay, I know zero about this domain. I know zero about this subject. So the more we read, the more we understand how to differentiate between a good research and a bad research. And once you understand that, you will be having the leverage of creating only good research and good methodological studies. Statistical skill sets. Now, statistical skill sets should not be limited only to software tools. Sometimes you come across, you'll like people, oh, I know R, I know Power BI, I know Microsoft Excel, or SAS, SPSS. It's not important that you know 100 number of tools or Python, like the latest things that are coming up. It's important that you understand the statistical the statistical policies, the statistical principles behind it, the Bayes theorem, like why should you go for a multiple linear regression versus a multiple logistic regression? What is the characteristic of data? And once you understand that, trust me, Excel is good enough. You don't need SAS or you don't need Python to analyze the data. Excel is good enough for the kind of data that is globally being used. Yes, maybe larger data sets, Excel might have a bit of a problem because Excel cannot really go well with very huge data sets. And when I'm saying very huge data sets, I'm mentioning around data sets of 1 TB or more, 1 TB, 2 TB data sets. But statistical understanding of the principles is more important. So invest time in that. Invest time in understanding what is OR, what is RR, how are they different, what is p-value, okay, when can you use chi-square and where is the limitation of chi-square ends. So those kind of understanding, and please study core statistical biostatistics textbooks for those kind of understanding. So next point, data visualization. So no matter how much homework is done, at the end of the day, the data visualization skills is of the key. That is how you communicate all your learnings, all your understanding to the person who is listening. And one of the major point to note here is to keep it as simple as possible. Your data should be palatable. Your data should be edible to the person who is going to ingest it. It should not be something that, too many graphs, too many data, too many different data points. But at the end of the day, you are not able to achieve your end objective. Your data should be presented or visualized in a way that it's very easy for a child to understand. So you can see these figures here. If you have seen some of the latest graphs that come out in most of the researches, they kind of fill the graph to a certain extent. Have I crossed my time? I'm sorry, just a second.
We still have a few minutes, Ananya. Please go ahead.
Yeah. Okay, sure. Yeah. So data visualization skills that come with it. So please make your data as easy and as understandable so that you don't even have to explain it. People can just look at it and say, oh, okay, this means this person is lesser than that. Okay, this relative risk is lesser than that. It should be that way, beautifully visualized. And report writing, scientific writing skills. Again, this comes from in-depth understanding of research methodology. The more you read, the more you'll understand the knowledge, that kind of language, crisp, as well as not too hefty on words, not very ornamental, crisp, simple language that a scientific paper demands. And the way you write it is almost similar to the data visualization, right? Again, writing a paper is also kind of communicating. So the simpler it is, the direct it is easier to go through international publications. I think I am at the end of my slides, so I'll just stop sharing. Any questions? Please feel free. In case you don't have any questions at the top of your mind right now, you can just write to us or write to Ashish, and you can go back. We also have Dr. Yannick with us, and I'm surprised, sir. Thank you for joining, sir. If you want to add something, because I mean the next session is going to be very interesting, and I'm also looking forward to it.
Thank you so much, Dr. Ananya, for being with us today and showcasing how connecting the dots around the unmet needs and collaborating with various stakeholders into the market, including the government, could impact millions of lives out there, irrespective of their socioeconomic status. Thanks a lot for that. We do have a couple of questions. So my first question to you is: in what ways do pharmaceutical companies engage in policy advocacy to influence healthcare policies that impact market access and health economics?
Okay, so I'll take this question, but Dr. Yannick, sir, if you're, please feel free to add in if I am inadequate in my answer. So this particular vertical is called government affairs. Sometimes it's called corporate affairs, government affairs, or corporate affairs together. What they do, again, there are firewalls. There is an extent to which a pharma company can involve itself, but the pharma company can always present its own research data. If I have invested my time and my resources into understanding the epidemiological need, the unmet need of the disease, the burden of the disease, that can always be put forward to the government, policy makers, the stakeholders, the decision makers, and you can always have a say. Pharma companies can also help patient access, patient access groups have a better platform, have a better voice. So suppose there are patients, there are caregivers of a very rare disease, and they don't know how to connect. They don't have a platform to connect. Uh, Pharma companies can always help them connect and help them raise a voice at the table when the stakeholders, the government bodies are involved. So that is one way pharma companies do it. But yes, there are firewalls. They kind of don't involve in every decision, because that will lead to a lot of ethics issues and ethic questions. Nobody does it in a wrong sense of interest, but they can always be perceived in a wrong sense of interest. So yes, scientific exchange is always allowed and is always welcome.
All right. Thank you so much, Dr. Ananya. My last question to you is: how do early access programs and compassionate use initiatives factor into market access planning?
Okay. So a early importation program or an early access program is a great learning opportunity. Not it it actually helps us understand the patient's journey so well. It actually helps us understand and kind of live and visualize the patient's barriers and challenges so well that it it basically becomes a setting stone. It becomes a first step into how you design your access programs. Because once, okay, the drug is not in the market, so the obviously patient is deprived of the treatment, but we know that it's launching, it's going to come, so we have to create initiatives to make it more accessible to our patients. But how do you actually get that learning from? It cannot be theoretical. Market access is absolutely anything but theoretical. Market access is absolutely on ground, real world studies. And so the next session is going to be of great interest to all of us, because real world evidence, patient reported outcomes, and their barriers, their challenges. So what better way than to have an example? What better way than kind of someone exemplifying it for it, and someone living through that journey, so that you design your solutions accordingly. So yes, there are very important programs, and most pharma companies kind of, if there is a need, if there is a expert who is aware of what is coming, which is the latest medicine that is going to enter the market, which is the latest kind of molecule that is going to have a new treatment protocol coming up, when they kind of recommend patients, all pharma companies try their best to have early importation or early access programs. I hope that I
All right.
answered. Yeah,
Yep.
thank you.
Thank you so much, Dr. Ananya, for being with us.
Thank
And
you.
thank you, students, for amazing questions, and Dr. Ananya for equally amazing answers around it. With that, let us move forward to yet another exciting session, and which is RWE Unplugged, Harnessing Real World Data Power. And for that, we have with us today Dr. Yannick Manouf, Director, Real World Evidence, AstraZeneca. Dr. Yannick, following a PhD in neuroscience from University of Manchester, UK, he worked in academia, pharma, and biotech industry in Cambridge, and also a consultant in London prior to joining AstraZeneca 10 years ago. He started in R&D oncology, where he provided clinical information support for key strategic imperatives, and then he moved to medical affairs 5 years ago, focusing on governance of real world data assets. With that, Dr. Manu, over to you. You're not audible, sir.
We won't be able to hear you, sir.
We are unable to hear you. No. No, not yet, sir. Not yet. While Dr. Anik is working on some technical aspects, let me request the students out there to put up their questions onto the Q&A section. It would be sent to us so that the speakers could actually respond it at the end of the session. No, sir. It's not audible yet.
Okay, now?
Yes, now it's audible. Yes.
All right. Sorry about that. I don't know what happened. Usually it's kind of seamless. But again, yeah, I was going to say thank you very much to the organizers for inviting me. It's a pleasure to take part in this session. And yes, so thank you, Anish, for the introduction. I work in medical evidence, and obviously I'm not an expert in real-world data, real-world evidence, but I work with a lot of experts in these fields. So I hope to be able to share some aspects of it with you. And first, as we had some problems with audio, I'm hoping that you can see my screen. Just can you tell me if...
Yes, Dr. Anik, it's visible.
Okay, so we have a go then. Excellent. So what I would like to say, sorry, that's not. First, the basics and some definitions regarding real-world data and real-world evidence. So real-world data are all the data which are relating to patient health status at any point in the journey, and which are related to the delivery of healthcare and are collected from a wide variety of sources. Okay, so that's that. The data is there. What do we do with this data? And that's the evidence part of it. Evidence means that you've actually used this data to generate clinical evidence on the use of potential benefits or risks associated with a medical product. Okay, so what you can ask yourself is, do you have access to real-world data? Do you use real-world data in your country? Would you like to run studies locally using real-world data? And do you have the capabilities to use this real-world data? As a snapshot, I think it's important to remember the difference between the data that comes from clinical trials and the data that is real world. Approximately 5% of all the healthcare data comes from clinical, okay? Only 5%. The 95% outside of this is real world data. So you can see the huge potential there is there in terms of gathering information, etc. So two types of real-world data. I'm going through some definitions here because I think it's important to set the scene to basically understand where we're going. Okay, primary real-world data. So that's the data that your company or your organization, here you've got the AstraZeneca logo, obviously, but it could be anyone in the field, is directly collecting by collaborating with external parties, with hospitals, with consortia, etc. But your company or your organization here is sponsoring the collection of the data. Okay, you're going out to get data. So that's primary. Secondary, observational real-world data is data that has been collected by a third party for other purposes by and by an external party, I mean, usually commercial, suffice to say. So the secondary data sets do not originate from your company. They are available, but they were not necessarily, the vendor will just have collected them and is providing you with a license to use this data. So that's secondary. You are getting data from the outside, but without having had a say in how it was collected in the first instance. Okay. Now, if we move on to the next slide, you might ask yourself, where does it come from? Well, if I mention the fact that it consists about 95% of all the healthcare data, you can realize fairly quickly that real-world data is everywhere. And I've tried to just highlight a few cases here from clinical, with everything that has to do with health records, electronic health records, and not EHR, sorry, yeah. Lab results, diagnosis, everything that has to do with digital. So it's something that is gradually coming on, all the trackers, all the devices. Again, it's maybe more prominent in some countries than others, something to bear in mind. The way the prescriptions are made in terms of orders, in terms of administration route, in terms of co-prescriptions with other drugs. The claims, very important for market access, medical and prescription. What's been coming our way as well recently in the past 10 years is all the molecular profiling, so genomic and genetic testing, if you want, and all the omics, so proteomics, metabolomics, etc., etc. An important aspect of also real-world data, I think Ananya has mentioned it, is PRO, so patient-reported outcomes. Is very, very important to incorporate all that you have regarding how patients experience the treatment and experience how their disease is evolving while being under this treatment. And actually something that is also very, very important now, if you're working in the corporate world, is social media. All the patient communities, patient groups who are on Facebook, on Twitter, all the blogs, etc. All this is really important in terms of how you want to understand how a patient feels. Okay, just put a caveat, is that this list is not exhaustive. There might be other sources that might be more relevant depending on the questions that is asked. And also, I'm not really, I must confess my ignorance with regards to the Indian kind of setup in terms of data. I mean, we have had some discussions with the Indian marketing company, but I'm fairly ignorant, and so forgive me for this. So when you look at this, all these examples here, some will be more relevant possibly to where you are in your state versus another one, for example. It's just the nature of how things are. Now, if we move on to the differences really between real-world data and randomized controlled trial in terms of data. Obviously, in a nutshell, one is very precise and confined, and the other one is very loose and wide. So RCTs, obviously, it's experimental. You're testing a drug on a very well-defined population. You have very stringent outcomes that you're expecting in terms of efficacy, safety, and obviously quality, overall quality of life for the patient. Again, the inclusion criteria are very stringent in phase 2, phase 3 trials, etc. So you will have a list of, as you know, of criteria, and patients who do not fall within this list of criteria will be taken out of the trial. Monitoring can be very intense in some cases. The cost is very high, and obviously it is randomized, and it is in theory, well, in the vast majority of cases, double-blinded, as you know. Conversely, real-world data is only observational. There are some interventional trials, but the vast majority of real-world data comes from observational, so just looking at effectiveness and not efficacy. That's the different kind of criteria here. But you're also looking at, you know, anything that would tell you, for example, appearance of new or different safety signals. So that should be also part of the primary focus sometimes. The patient population here obviously is completely unrestricted. It's everyone is included. There's no monitoring required. The cost is less, far less in general, as well as the patient population will be much higher. So a phase 3 trial may include, let's say, 1,000 patients. Okay, it's possible in real-world data to get millions of patients included. So there are differences there as well in terms of patient population. Randomization, yes, there's no randomization at all, because you accept, obviously, you get data from all the patients, regardless of any kind of criteria here. So from a kind of scientific clinical perspective, there are some advantages, but you also have to think about at first it's testing the drug. So you have to mitigate, basically. Think about it. How can you test a drug using real world data? Will that work? So this is where, yes, real world data is very useful, because it will help you understand how the drug works better, and possibly refine how the safety aspects are profiling themselves within a much wider population. But at the same time, it's fairly difficult to go without randomized controlled trials. So bear that in mind. In terms of the quality of data, that is a really, really important aspect, which is a big stumbling block in many countries, because you need to have data that is of high quality to be able to assess a particular question. And that is not always the case, just because it's collected in different types of ways, and people may or may not incorporate the same type of stringency in how they collect and enter the data. So you need to have completeness, which is the amount of data should be complete. You should not have gaps, basically, and that happens quite a lot actually in real world data. Accuracy, so patients should have the same characteristics in terms of how the data is collected. Timeliness and representativeness, when was the data collected? Where was it collected? Is that representative of a particular disease, population, etc.? And comparability. So it's also another term called interoperability. So how will this particular set of data, how will you be able to compare it to another set? It's really a complicated issue here, because you can imagine in India itself, there might be hundreds of thousands of registries available. So are all these registries, packaged using the same type of software and the same type of coding? Go outside India, go to China, go to Japan, go to Europe, go to the US. You can imagine that over the world, this interoperability is really a big problem, because there isn't really a standard model that people use. So it's something to really bear in mind when you're thinking about doing a study and maybe setting up some big databases there. Okay, let's move on to the next. Uses for drug development, for medicine development. It's moving into all aspects of drug discovery and development. From the research, it can enhance preclinical, yes, knowledge, absolutely. Strategy, trying to define better the target product, target profile of the product. That's also possible. The way to design clinical trials as well can be influenced. It's happening now, and patient insights. So that's what I was saying. It's something that is taken. Think about it. I was going to say, if you have that in mind, to try to set up a real world evidence piece. Think about patient centricity, competitive insights, commercial insights, etc., execution excellence. I mean, it really is. The only aspect maybe that is, I would say, less obvious here is regulatory. I haven't done a slide on it, and if you go to the big regulatory global organizations such as the FDA or the EMA, they would say that yes, they're perfectly happy to consider real world data and so forth, and it's important to have it, and they've published guidelines on how real world data should be presented. If you decide to have a cohort, it happens. Again, I haven't done a research on, or I haven't presented slides on use of data in the literature, but in oncology, for example, it happens quite regularly now that you have control cohorts which are coming from real world evidence, real world data, just by the fact that obviously from an ethical perspective and also practical perspective, it makes more sense to have that already, to have it a cohort that's already there, so that happens there in the real world. But at the same time, it says that we are there to the two are compatible. The two are there to support each other, but one cannot take over the other one. So real world data is there to support clinical trials in making the drugs better, in defining how patients are experiencing the drugs, are experiencing side effects related to the drug, and how we can mitigate this in the phase 4 life cycle of the drug. Okay. In terms of specialist users, so life science researchers, and that's very broad. So that goes from preclinical to clinical research. It's everything that you get from a very, very wide population with multiple. That's the difference with the clinical trials. Basically, you don't have a very narrow band of patients. Those patients are coming from all sorts, all different, ages and so forth, and they have different comorbidities, they have different treatments, etc. So you get insights from all this, and it's really important to be able to say, oh, there's something going on there we haven't seen before. So it's really, really a mine for all sorts of insights in terms of figuring out how your medications are working. And also market access, it's key to be able to, if you're going to the payers and you say, we've got this wonderful drug here, and we would like to sell it in the country at this price, and they're going to say, well, show me the money. How do you think it's going to be worth that price? So this is how you're saying, well, we've got, we've got the results of an observational study here, which shows that the drug actually reduces the side effects gradually in a much more pronounced manner than a competitor, for example. So that's a way that you can influence, if you want, how the market access will happen. Also, if you have the proof that this drug generates overall far less or significantly less side effects, or is better tolerated, and so forth, then another one, even if efficacy is quite similar, that's a way to influence market access. And for physician, I think here it's related to life science as well, but it really is at the high level. You think about a drug, you think about a class of drugs, and you're saying there are differences, and this is how we're going to make this drug better, by looking at the overall picture and taking into consideration that this drug has maybe 20-30% less side effects, while the efficacy is absolutely identical. And that's how the regulators, that's how the health authorities decide that, overall you're gonna get maybe preferred access to a particular reimbursement. Move on to the next slide. Now, I'm going to talk here about real-world data selection, and this is particularly related to secondary data, because that's what I know better. So I thought to just, if you ever in a kind of corporate environment, or you have you're in a big organization which has funding, and you say, okay, which company in India is actually providing data? So first, you need to determine what you want to look at. You have a business need, you have a scientific question, and which data are you going to be able to work on? So the variability is largely unknown. That's true, and also very unlikely that a given commercial data set will be a perfect match to be able to answer your question. So it's very important to consider that, because remember that those commercial data sets were not collected with your need in mind. It's just something that is available, and you have to accept that there will be some good and maybe some less good within. So you approach a set of vendors, you have a set of criteria to test the data closeness to your needs, And you have also a way to score the best option in terms of quality, in terms of cost, in terms of whatever you need to be included in the criteria, selection criteria. Then you have the procurement, licensing, ensure appropriate availability. You need to have data storage, make sure obviously that the patient privacy laws are absolutely respected. Usually all these data come de-identified or anonymized. And then you have a data set lifecycle within your organization. You have a license for a year or 2 years, etc. And that's how a lot of, I'm talking about a pharma organization here, that's how we generate most of our evidence, really. It's using those commercial data sets because they are used by many people. They are even cheaper than actually going and generating your own primary observational data. So, final comments. There is someone at the origin of real-world data, and it's usually a healthcare provider, could be a physician in a hospital, could be, health authorities locally. It varies, but someone must enter the details regarding the patient X, for example, into an electronic health record. Otherwise, there will not be any real-world data. So if ever you're in this position, think about you have a very important role to play, because without you, going onto your keyboard and typing in the details, nothing will happen. As I said, real-world data studies cannot be a substitute for randomized controlled trials. They can support many aspects of how a drug will be prescribed, will be reimbursed, will be authorized at global level, but it will not substitute the very precise indications and information that you get from randomized controlled trials. Again, keep patients in mind when you start looking at potential studies. It's really, really important to have patient outcomes in mind. You have to know your local real-world data landscape. Again, I'm sorry, I do not know much about what's happening in India, but it's a good exercise. If you ever have some time, or if you have a project, look at what's available out there. It'd be great to know, are there commercial vendors? Are there registries available in Mumbai, in Delhi, and wherever? Are they talking to each other, etc.? And also, I think the audience is medical. If you embark on real-world data / real-world evidence generation, you need epidemiology, data science, and statistics capabilities. So it's not something that necessarily just comes out of a shelf. You need to employ the right colleagues there to help you generate this. There are also companies that provide this kind of service, but you need to have a budget for it, basically. Okay. It's very general, very much of an overview, and I'm happy to take any questions. If we don't have the time now, please liaise with Ashish and the team, and I'll be more than happy to answer any questions offline.
Thank you so much. Thank you so much, Dr. Yannick, for being with us today and sharing your critical insights around real-world evidence. We do have received some questions from the students. So my first question to you is, how are emerging technologies such as AI and advanced analytics being employed to extract meaningful insights from vast real-world data sets?
Yes, it happens, but the problem still remains. You may try AI in a particular setting and that might work, but it's not applied globally, and that is the issue. It's the same issue as the actual interoperability of the data. You need to have a global consensus, which currently is not happening. There are yes, there are studies here and there using AI which show that it's potentially very useful. I'm not saying it's not, but the problem is not everyone is using it, and not everyone is using the same AI model. So it is useful, and it shows possibly a direction in terms of how the data will, in 10, 15 years time be processed. But at the moment it's a bit haphazard, if you want. So but yeah, definitely. I'm not saying it's not it's not happening. It's happening, but not quite there at the moment.
Thank you so much, Dr. Yannick. My second question to you is, what ethical considerations should pharmacology students keep in mind when working with real-world data, particularly when it involves patient information?
Okay, yeah. So you, like in any trials, you will have informed consent forms, when collecting the data. So if you're working in a hospital, you will have 10, 50, 100, 200,000 patients, but they will all need to have signed an informed consent form saying that, they're happy for it. Obviously, it's not a trial because there's no, you're not giving them any drugs, or there's no treatment per se, but you're using their information. They need to know that. And also, if you publish, you need to mention that, or if you use the data, it needs to be anonymized or de-identified. You need to have a way at the source to remove all these names, addresses, phone numbers, email addresses, etc., before you can use the data. Okay.
All right. Thank you so much, Dr. Yannick. My last question to you is, how important is collaboration between different stakeholders in healthcare ecosystem for successful utilization of real-world evidence?
It's key. It's absolutely key. And I work a lot with international, and it's absolutely mega important, just by the fact that we're not a one nation environment at the moment anymore. We're a global environment, so you need to be, from the state of Gujarat in India, you need to be able to talk to, the state of Victoria in Australia. You need to be able to talk to your colleagues in Japan. You need to be able to talk to colleagues in Germany. Anything where you have a question, where you think about an idea, think about outside the box. Okay, you have your local setting. Think regionally, think nationally first. But then it's perfectly okay to go outside and say, has anybody done this kind of study? Has anybody done, thought about doing this? And if you have someone that says, yes, it proved really successful. So absolutely, collaboration is key here, and not just, think outside the box, outside your local setting.
All right. Thank you so much, Dr. Yannick. Thank you so much to the students for asking excellent questions, and Dr. Yannick for being with us today and patiently answering to the questions which are coming across from this. And thank you so much.
Thank you, Anish. Thank you. Bye.
Thanks. So with that, let us now move forward to our next session, which is pen to paper mastering pharmaceutical publications. And for that, we have with us Dr. Chinmay Joshi. Dr. Chinmay is a medical graduate with a postgraduate diploma in clinical study management and around 15 years of experience in medical writing. She started her career in medical writing after working for a couple of years in preventive and social medicine. As a writer, she has developed technical content for oncology publications, and after being with Fortria Scientific Private Limited for more than 9 years, her role has evolved from medical review of regulatory and publication documents to manage a team of medical writers and reviewers and account management. She sincerely appreciates and enjoy the part she gets to play in disseminating cutting edge scientific information to the fraternity of healthcare professionals. With that, let me now hand over the forum to Dr. Chinmay for taking it forward. Dr. Chinmay, over to you.
Thank you so much, Ashish. So let me share my screen. I hope it's visible to all of you.
Yes, yes, it's visible.
Okay. So I have actually two observations about the topic itself today. First is about pen to paper. So actually in today's age, it should be keyboard to screen. There goes my lame, attempt at comedy. But yeah, to make it more relevant, it should be keyboard to screen now. And second, on a more serious note, is mastering pharmaceutical publications. So honestly, with the time that I have spent, a decent number of years, more and more, I realize that it's really not possible to master pharmaceutical or any kind of publications as such, because we realize that the more we work with a form of art and also a science, the more difficult it is to master it. We all are attempting to learn, we are all attempting to share, and we are all attempting to do the best we can. But mastering seems to be very difficult. So just on a lighter note, but yeah, to begin with, I wanted to just share my realization with you guys. So thank you so much for joining and having me today. Thank you for inviting me. It's always nice to talk to young minds and see what newer and brilliant ideas they are coming up with. And I consider this my privilege to be with all of you guys today. I'm also a little conscious or of the fact that I am actually following up such stalwarts of the industry. But whatever I have learned along the way, this is, I would think this is would be a great opportunity for me to share it with you guys. So yeah, let's begin. So I would just like to take you on a brief journey actually as to what is medical writing, specifically medical publications and pharmaceutical publications. What do we actually mean by them? How do we come up with them? What is a good way of presenting them? And what are the career opportunities for people like you who would be interested in a career in medical writing or publications? So yeah, I mean, to begin with, starting with the fact that billions of dollars go in research, by pharma companies to bring a few products to the market and essentially to the patients. So the ability to convey the results of this research and development that is going on is basically publications. The way we convey the results of the ongoing research to the end users in the healthcare industry, let it be payers, let it be patients, let it be physicians who prescribe the products. It happens via medical publications. So basically, as I know, the first point says that they are cornerstone for driving innovations, sharing, knowledge enhancing, and basically coming to the foundation of it all is enhancing patient care. We are grappling with so many conditions known and unknown to man. The COVID pandemic was a classic example which had the entire healthcare industry scrambling on their feet, and they stood up to it. But yeah, I mean, newer and newer challenges are coming every day in front of us, and how we tackle them and how we share our experiences within the medical fraternity or within the healthcare fraternity with each other, how we are able to combat these things and how we are able to get the required product or the required care to the actual person who needs it the most. That is the way which happens through medical and clinical publications. So they lay a foundation for evidence-based medicine. We, as I said, that we communicate the results of clinical trials, through these medical publications. This is evidence, that can be shared with the industry, that can be grown, that can be worked on, and that can be enhanced as time goes by. These kind of publications are also a good medium by which people who are already practicing medicines get to be abreast of all the newer things that are happening in this industry, the newer innovations, the newer products, the products that can help them treat their patients more effectively with drugs that are more safer, which will cause lesser adverse events and essentially give a better quality of life to their patients. So this is a very, very common way in which you can actually convey the latest cutting edge, knowledge to all the physicians practicing and helping their patients. This can be also a very good medium for patients themselves. As we all know, patient advocacy groups are now being encouraged. A lot of pharma companies are coming together and actually making patient or basically encouraging patients to come together, seeking guidance from them as to which newer or which medications can help them better combat their conditions. So these patient advocacies groups have now evolved, and they are now encouraging patient authored medication publications also. So what happens that one of the authors of the entire publication is a patient himself or herself. So that narrative gets added to the publication, and this in turn helps the entire healthcare industry to understand the condition from a patient point of view, the condition, or maybe even the product profile. Like if you're talking about a particular molecule, it is so common for us, or so nonchalantly we say, the common adverse events associated with this molecule could be nausea, fatigue, vomiting. Just an simple example. But if a patient adds a narrative to this publication saying, we are reporting, for example, a case study, and we take the verbatim from the patient and add their narrative to it. My nausea was so bad that I could not eat the entire day. I had such severe vomiting that I could not even reach the washroom, and I had to clean up after myself. I was so fatigued that I could not take my children to school or nor play with them. Doesn't it add a whole lot of a different perspective, to a simple publication or a simple case report that we might present about an adverse event profile of a drug? So this is what happens, when medical publications actually involve the patient themselves, and this can be a very empowering medium to do it. And then when we have enough evidence, when we have enough practitioners on board, when we have enough researchers on board, this can be a very good medium where you develop a collaboration between people and you can actually influence healthcare policies. So we developed a consensus document where there was a panel of medical oncologists and Indian medical oncologists, and this was about screening of lung cancer. And we have policies of screening of lung cancer in India, but this panel came together and obviously they had very good recommendations, and they were all very experienced. They had very good recommendations, and they came up with an expert consensus statement, where which was used actually to influence healthcare policy or screening policy of lung cancer in India. So these things are a reality. They do happen, and we get to play a vital role in all of it. So it is a very gratifying, I would say, industry to be a part of, where you understand that what could be the significance of the work that you're doing. So yeah, who all needs medical writers, or where are the places where medical writers or publication specialists could work? One is obviously the pharma industry, where the research is taking place and they would require people to communicate this research to the healthcare industry, to the regulatory industry, to the physicians, to the payers, everybody. Clinical or contract research organizations like I am a part of, where on behalf of the pharma company, we do the documentation either to the healthcare authorities or to the publications. So then scientific and healthcare communication companies, where they do just the communication part, and then media houses and public publishing companies, healthcare websites, and digital platforms. This is a very, very new field and very rapidly evolving and very extensively used nowadays by healthcare professionals and practitioners. So that is also a very good medium where medical writing and medical writers could make their mark. Okay, so going a little bit into the depth of what medical writing encompasses, it's essential to know that medical writing overall is a science as well as an art, because art, because it requires a skill, or I would say even a passion for communication, and it requires that the reader actually gets interested in what you are trying to say, or what you are trying to convey, what you are trying to communicate. So holding the interest and then staying authentic to it through scientific knowledge. So that are the two basic requirements for medical writing, or good medical writing, I would say. The 3 C's, as we say, for medical writing, essential 3 C's are content, clarity, and compliance. So content, obviously, scientifically, you have to be 100% accurate. There are no two ways about it. And you guys being from medical background, you're already halfway there. So you have the proper knowledge, because sometimes what happens is there are medical writers who may not be from medical background, obviously, but extremely interested in having a career or starting a career in medical writing. Unfortunately, they might lack the scientific expertise or the understanding of medical or clinical concepts. So preferably people from life sciences background only should go into it, or can go into it, I would say. But being from medical background definitely is a big advantage. Then clarity. I think this is the basic ingredient of any effective communication, because unless and until you yourself are not clear about what you want to communicate, the reader or the receiver obviously is going to get confused with what you have to say. So content, clarity, and compliance. So when it comes to writing, there are obviously there are I would say chances, or there are requirements at times where you could take your material or borrow your material or get inspired from certain other publications or certain other places, where you have to acknowledge from where you are referencing the material that you have taken from. You have to be completely transparent and avoid plagiarism at any cost. So content, clarity, and compliance, this will be reiterated throughout your career if you choose to embark on this part of medical writing, that these are the essential components. And then when we started off, it was ABC, accuracy, brevity and clarity. So even that holds true even now, and as I said, non-negotiable. Okay, so so these are I would say, the traditional types of medical communication documents that you would come across, and probably some of you already know, or most even. So these are the actually type of publications that are seen most commonly. So these are original articles where there is a clinical trial going on, and or an observational study, and then you publish the results of that study. That is an original article. A review article is where you are talking about maybe there is a new molecule for example, in oncology. So you are not talking about a particular clinical study, but overall the place of the molecule in the therapeutic area, how different it is from its predecessors, what are the efficacy advantages, how better it is in terms of its safety profile. So you will give a background of the therapeutic area, you will give the different molecules that are used, and then the place of this molecule compared to them in that particular condition. So this will go as a review article. Then there are conference abstracts. As we know, there are various clinical global societies all over the world which have their conferences mostly once a year, and wherever newer research or upcoming research needs to be presented to the clinical fraternity, this is a very good platform to do so. And a very crisp and concise way of doing it is, you present a conference abstract. Then there are systematic reviews and meta-analysis where you actually work together on the clinical results of various clinical trials, and then you present a composite result of its own. So those are SRMS. Pharma companies end up sponsoring a lot of SRMS to again showcase the efficacy of their product among other products in the therapeutic area. Clinical practice guidelines and other types of publication where again a panel of experts comes together and discuss the latest research for a particular ongoing research or latest published results about the use of particular molecule for a particular therapeutic area, and actually guides the healthcare industry, or guides the physicians as to what should be the first line treatment, the second line treatment, or the third line treatment for a particular condition. So those are called clinical practice guidelines, and they are published as well from time to time. Either newer guidelines for newer conditions. When it came to COVID, we had a deluge of clinical practice guidelines being published. If not as extensive, for established conditions, always there will be updates. Or some field like oncology, where newer and newer products are entering, or the products that were used in as a part of as for patients only in later stages, and they are finding evidence that this could be helpful for earlier stages as well. So these guidelines get updated from time to time very frequently, and they get published. Consensus documents, as I said earlier, like an expert panel come together and they give recommendations. Sometimes the recommendation can be about how to adapt global guidelines and locally. So the conditions might be different, like obviously in Western world the conditions are different, and the way a clinical practice guideline could be adapted to a country like India, there could be many tweaks that are required, or many local considerations that could be required to actually implement those guidelines at a regional level. So these kind of consensus documents are also published for the regional audience. So in short, I couldn't go on and on about this, but in short, these are the types of various publications that we are used to writing. And then, like for any other scientific process, there are guidelines available of how you do that. Where if you are publishing an original article, there are very comprehensive guidances available throughout. And how do you present an original article? How do you present a review article? How do you write a review article? Everything, what you can see on the screen, every type of publication has a guideline that you can follow, and you can write an article as per the journal requirement and submit them. Then there is a committee, International Committee for Medical Journal Editors. They also, and this is the most commonly followed actually recommendation by all writers to write any kind of article. And again, very comprehensive set of guidelines are available, where even a non-writer, I would say, would find it easy to convey what they want to convey, because they encompass very minute and detailed instructions of how to write each and every section of whatever article you or whatever type of manuscript you want to write. So we'll just, as I said, mastering is not possible. We are just skimming the surface here. But if done properly, maybe it will pique your interest and hook you to this field, as I have been hooked for the last several years. So getting a little bit into details of how these guidelines can be very specific and what do they actually tell us. So the title, everything from the title to the conflict of interest information that is submitted for an article. Guidelines are available for each and every section, subsection, as I said. So, and there is a lot of thought that goes behind writing these particular sections and how to present the information that we want. One is to transparently convey the scientific information that we intend to, and second is to catch the interest of the reader. So the most essential aspect, I would say, is to understand the purpose of writing that particular piece of medical communication or clinical communication that you want to write. So if the purpose is clear, if the intent is clear, it becomes very easy. And so if you're writing if you're writing an introduction for a protocol, for example, a clinical trial protocol, that this is going to be submitted to a healthcare authority, where a regulatory person is going to read it and try and understand the significance of the clinical trial that you intend to conduct. If you're writing a review article, for example, which is meant for physicians, that you need to give the introduction in such a way that how is this piece of information going to help them treat their patients in a better way. So the audience has to be kept in mind, the reader has to be kept in mind when you are writing any type of article or any piece of medical communication, I would say. So the title identifies the article or the manuscript that you want to write. Again, since it is writing and it is communication and it is an art, but it is scientific art, you have to keep that in mind. So filler words are actually discouraged, like buzzwords are actually discouraged in any kind of medical writing. It has to be crisp, it has to be to the point, and it has to be scientifically valid. Those are the essential parts, and of course interesting. So title, as your name identifies a person, the title identifies the manuscript or the article that you're writing. Very important part, and it immediately can hook a reader to the content that is coming next. Then the abstract. So abstract is when you publish or when you send a manuscript to a journal to assess whether it is okay for getting published. The abstract is the piece of information that the journal editors look at first, and then on that basis they usually make their decision whether to accept or reject the article that you want to publish. So it has to be catchy, it has to be crisp, it has to be concise, and it has to convey the main idea of your manuscript or review article or any other type of article that you are writing or you wish to write. Sometimes you also have to bear in mind that sometimes abstracts are the only freely available parts of the article to many readers. So, and if they have to read the entire article, they have to pay certain fees. So the article should be the abstract should be complete in itself, even though if the reader does not get to read the entire manuscript, only by reading the abstract, he or she should understand what you are trying to convey. If it is a original article, the gist of the results should be there. The entire gist of the results should be there in the abstract. If a review article is being written, the abstract should mention what is the value of the message that you wish to communicate. So the reader should not be left hanging just because they can't read the entire manuscript. They should not be left unattended, that okay, this is what it is, you will not be able to read or you will not be able to understand what is there in the entire manuscript. No, the abstract should be self-standing and self-sufficient. So this probably is a very well-known form in which probably all of you have written papers already, the IMRAD form, where we know that there is a particular structure, and we generally try to follow this structure even for review articles at times, not always possible, but introduction, methodology, results, and discussion. This is like the core of any manuscript that you would want to write, and this is followed like a Bible in any type of medical writing communication that you would want to do. So before moving further, I just wanted to discuss this hourglass figure with you, where we encompass all the sections and probably some subsections also of a manuscript. So we start with the title, the abstract we have already covered. The introduction, it's like a funnel shaped thing coming on. In the middle is methods and results, and then discussion and conclusion, ending with acknowledgements and references. So this is the entire structure of a manuscript, so as to say, and the shapes have significance. So moving on, when we discuss probably the subsequent sections, we will come to that, but the shapes of this figure also have a significance, starting from introduction. So the purpose of writing an introduction is giving a background to the topic that you want to discuss. So as the shape of the funnel, we start with what is known. So for example, if we want to write about a fairly common condition like diabetes, for example, so burden of T2DM, we would start with that, and then slowly move on to, if we are in our research, if we maybe are talking about one particular interventional drug which is known to reduce the rate of cardiovascular complications in patients with T2DM. If we want to write about, then we would directly start with the rate of the burden of cardiovascular complications in patients with T2DM. So depending on what the topic is, depending on what you want to write about, we start with that. So what is known? And then we come to where is the gap, what is not known. So if again, going back to the same example, we don't know how this intervention is going to reduce or may reduce the rate of complications in this particular patient subset. We say that we know that the burden is very high, but in a particular subset of population, like maybe an early onset T2D, we don't know, how this molecule could work, and we are trying to find that out. So what is unique about this study, and what gap in knowledge or what gap in medical knowledge is the study going to fill? That is how the introduction should end. What we know to what we don't know. So a funnel shape. Then methods, like how did we conduct the study? So we generally have the clinical trial protocol before us when we are writing the methods section of any original manuscript. So I would say the most essential component of any method section is transparency. You have to basically be 100% transparent in what was done in the clinical study. Do not try to unnecessarily exaggerate or hide anything, in the methods section. And basic requirement of this study is that if somebody wants to replicate the same study design, by reading this methods section, they should be able to do it. It should be that detailed and that I would say elaborate. Yeah, it's the same thing, but it should be that detailed that a person wanting to replicate this study should be able to do it by reading the methods section of any original manuscript. So a very good way to do that is creating flowcharts and diagrams. So, generally a well-written manuscript will have good flowcharts, will have one or two diagrams to make, reiterate some points or present them in a better way or make it more visually appealing. So methods section and also to, be compliant to the word count. So journals have very strict word count limits, and to remain compliant to them also, these things can come in very, very handy. So we moving on to the results. So we did introduction, we did methods, and now coming to the results. So again, results are the heart, I would say, of any original manuscript, and good results speak for themselves, So, and it doesn't mean that your product is shown in a better light compared to placebo or a comparator drug. No, good results can also be that, it was not as good, but a study conducted nicely and the results, which are presented according to what happened, and if we are able to convey the entire story, you need not then explain it further. That was the point. So they should be able to, I would say, speak for themselves. So we for a original article, we start the results with patient enrollment. Again, a flow diagram comes in very, very handy for patient recruitment figures when we want to present. Rather than writing everything in tedious texts, it's very, very easy to show how many were enrolled and then how many were excluded for which reasons, and then which actually what number became the study patient population. So patient enrollment, socio-demographic characteristic, these are very basic things. Clinical characteristic, and then you have to be very particular about which TA you are dealing with, which therapeutic area you are dealing with. Obviously will differ with different TAs, and then the knowledge of clinical presentation of a particular TA becomes very handy in these cases when you know that what kind of clinical characteristics would be a reader actually interested in. So if it is an oncology publication, that a reader reading these results would want to know how many patients had cranial metastasis, for example. They would want to know this. So you know that you would want to have these kind of clinical characteristics details in your results section. So again, depending on the reader, depending on the topic, depending on the in-depth understanding that you have about the topic, you can decide which information you want to present in which section and how. And then obviously the primary and secondary outcomes of the disease. Again, you have to have in mind what would the reader want to know. Would the reader if again, I'm coming to oncology because this is the most ongoing, I would say, topic nowadays, and people read about it a lot and want to write also about it a lot. Newer and newer drugs are coming, for treating these conditions. So what would be the reader interested in? Whether the reader would be interested in a better safety profile compared to the choice he or she has right now, better quality of life for the patient, a quality or even in palliative care, what would be the most suitable drug to give a better quality of life to the patient. You have to understand that, like, what would the reader be interested in, and what would be the details of the results then would look like. So we have to start from there and then move on to the details that we want to include. So we have to show a lot of integrity actually to the topic that is being discussed and the reader that is going to read what you have written. Okay, so here are some do's and don'ts about figures and tables. One basic thing, I mean, the details can be read anytime, but one basic thing is whatever you have presented in a table, do not duplicate it in text. That is a very basic rule that we all try to follow. Again, one is it becomes too tedious for the reader. Whatever is there in the table, why would you again want to read in text? And secondly is to remain compliant to the word count. So it becomes very easy when, and sometimes visually, it becomes much more easy to understand the data than to read the text. So again, whatever you have put in that text, don't write about it. And secondly, if a person reads or sees a table or reads a figure, looks at a figure, it should be self-explanatory. Whatever abbreviations are being used have to be explained in footnotes. So you don't have to again scroll through the manuscript to see, what is this? What is that? I can't figure out. So sometimes abbreviations are not that common, and the reader won't be aware. What is PFS? So better to explain it then and there on the footnote. And very commonly it happens with people from medical background who have come in the field of medical writing. They assume that what they know, all the readers might also know. So, the technical jargons, the abbreviations, we have to be actually very careful of that, you explain them properly at the right places. Okay. Then we move on to the discussions. So this discussion is nothing but actually seeing your information in the light of other similar information that could already be available. So how does this new piece of information fit into the information that we already have? Is it in line with what we already know? Have other researchers also found out the same things? Is it somewhat different? Did I deal with some different subpopulation where the same study or similar study gave a different result somewhere else? Is my patient population ethnically different than the other populations that were studied, and therefore my results are a little different? So especially when we are maybe testing for example, testing the prevalence of genetic mutations. So this is very common, that the prevalence of certain mutations are common in certain ethnic groups. So then the discussion section can actually become very interesting, that what was the prevalence found in Caucasian population, what is it in Asian Pacific population, what is it in African population, and the intention of doing this is how could different products help these populations in a more effective way, because we know that certain products or certain molecules are more effective in people with certain mutations, or less effective also at times. So what could be the best way of helping these patients? So intention is that, and then when you compare the results, when you discuss this scientific information in the light of what is already known and what could be the implication moving forward, this becomes a very comprehensive and interesting piece to read. Discussion section also generally ends with strengths and limitations of the study that we have written about. So strengths, people don't find it very difficult to write, but limitations, at times, yes, it is difficult to admit that my research did have so and so limitations. But again, in the interest of transparency, we have to have complete integrity to what we have done. We have to actually say that this could be the limitations. The smaller sample size could be a limitation of the study, or only a certain subset of the population could be a limitation of the study. Excluding a certain age group from the study could be a limitation. So until recently, actually, geriatric population was never included in oncology trials. And we know that the prevalence of cancer is very high in geriatric population. So now there are newer guidelines, the diversity and inclusion criteria are making pharma companies more and more vigilant about including all these patients in their clinical trials. But till recently, a lot of marginalized population would never be included in clinical trials, which could be again a very big limitation of any clinical study. So this is how it goes. We have to admit where the trial fell short or the study could not encompass, what could it not tell, and then the conclusion should say about what could be the impact of this knowledge. Is it opening new vistas of clinical research? It is reiterating what we already know, and should it be included in some clinical practice guideline? What could be the impact of this knowledge that we are trying to convey? That should be included in the conclusion, and that is what we generally try to do. Okay, so again, do's and don'ts of discussion, I think we have already done that. Then we come to the references section. We know we have to reference each and everything that we include in our writing, and different softwares are now available to help medical writers do that. And it is basically for the purpose of acknowledgement that we have referenced this material, and this is the basis of our discussion or whatever we have presented in the introduction. This is where we have got our statistics from. Plagiarism and copyright I did not explain in detail. We can't do without it. Copyright, yeah, if you are actually maybe even recreating a figure that you have seen in some article, or if you have maybe referenced NCCN guidelines, for example, in your publication, you have to ask for their permission that we are using your guideline as a reference in our article. Can you please give us the permission to do that? That is basically seeking copyright permission from the references. And just to summarize, we have everything here. I am already now what we discussed earlier. I know we are towards the end of the time that was allocated to me. So this is self-explanatory, and I have basically talked enough about each of them. So just wanted to leave you with this career graph, that very career graph, interesting one that I found in one of the publications that I referred to. So anybody from medical sciences, I would highly, being passionate about this path of career myself, I would highly recommend it to anybody who is good in communications and wants to be a part of this ever-growing industry. So two pathways are there. Writing, and you started as a writer probably because you're all medical graduates. You could start as a medical reviewer, grow on to be scientific directors, and sky is the limit. You could end up being president of the company that you work for. And if you are interested in SME kind of role, you start as a medical editor actually, and you review the content from medical point of view, and ascertain that it is medically accurate, whatever is being written. So these are the two career pathways. It could be in a pharma company, it could be in a CRO, it could be in a publishing house, if you are really interested in writing exclusively. So this is what I wanted to share with you guys. Thank you so much for your patient hearing.
Thank you so much, Dr. Chinmay, for joining us today for day 2 of PharmaSteer 2023, and at the same time taking the students through 360 degrees of the publication, which I'm sure that would benefit the students out there. Thanks a lot for that. I do have one question. I know that we are crossing the time limit. However, let me just take your quick comment on one question. How do researchers determine the most suitable journals for their pharmaceutical publications?
Good question. So we do a lot of research actually before we decide on which journals to approach for our articles. First, because we are a CRO, it depends on the preference of the sponsor company where they would want to publish, the impact factor of the journal, the range of impact factor in which they would want to publish. Third would be a global journal or a regional journal. So we narrow down on the basis of that. These are like the 3 criteria that we decide on.
All right. Thank you so much, Dr. Chinmay, for being here and responding to the question. Thanks a lot for that.
Thank you.
This brings us to the last but not the least part of the agenda and agenda for the day, and which is significance of pharmacology in drug development. And for that, we have with us Dr. Julius Vard. Dr. Julius's career has expanded across various roles in pharmaceutical industry in both early and late stage drug development, medical affairs, clinical operations over last 3 decades. He retired from Novartis as Global Drug Development India in June 2021, where he was Senior Global Program Head, Vice President Global Health, responsible for cardiovascular, metabolics, oncology, and women's health portfolio in global health. During his journey, he has led large teams in development, being based out of India, Denmark, Singapore, and Canada. Dr. Julius is a physician by profession and has received his MD in pharmacology from St. John's Medical College, Bangalore, India. In addition, Dr. Julius also has diploma in pharmaceutical medicine from Royal College, UK. Dr. Julius is presently guest faculty at several academic institutions where he gives guest lectures to undergraduate and postgraduate students in medicine, pharmacy, and allied specialties. He's passionate about learning and building skills and capability across academia and industry. He has over 30 scientific publications to his name. With that quick intro, let me hand over the platform to Dr. Julius. Sir, over to you.
Thank you, Ashish. Ashish, I'm trying to present my screen. I'm not seeing. Can you see my screen?
Not yet, sir. Nirav, can you please help Dr. Julius?
So still not. We cannot see the screen right now.
There is not. There is. I'm not getting a thing to show the screen. Give me a minute because I'm not getting.
Yeah.
Earlier it was
Yeah.
there.
Okay.
Now it is not there.
You open the presentation, sir, first?
My presentation is open on my laptop right now.
Okay, okay.
Nirav, can you put up your email ID to Dr. Julius so that he could transfer? Maybe you can share from your end.
Yes, sir. Yes.
Yeah. Dr. Julius, is it okay for you to send a slide deck to?
Yeah, no problem at all.
All right.
I can do that. Earlier it was coming screen show. For some reason, it's not coming now. I'm a bit confused. Okay, 1 minute. Let me just try it.
Yeah.
Can you see now?
Yes, sir.
Yes,
Now
sir.
it's visible.
Yes.
Okay.
Just put it full screen, sir.
Okay. Is it clear?
Perfect. Yes, sir. It's visible.
Okay. So to begin with, let me thank Ashish and the Astra team, but also the KEM team for making me, inviting me to present on the significance of pharmacology in drug development. I must, to begin with, I must admit, given that there are so many MD students online, I did my MD and finished my MD way back in early 93 in pharmacology. And honestly, I did not have a path forward at that time. It was like entering a dark forest. 30 years later, I must admit, the journey I've had through that forest has been completely amazing. And why am I saying that to you today? Is that because the opportunity that you have in pharmacology is significantly more than what I had 30 years ago. You have to seize that opportunity and make the best of it in your career. I want to begin with this slide, which talks about the average global life expectancy. A while back, I was just reading about the average global life expectancy. And I was surprised to read that if you were born in 1900, your average global life expectancy was a mere 31 years. Now you fast forward that by 120 years, just prior to the onset of the COVID pandemic, life expectancy increased to 72.6 years, more than doubled. Now the question is, I asked myself is, what has changed so dramatically in 120 years that has not changed in 200,000 years? And why do I say 200,000 years? Because if we believe in the evolution of man, modern man is believed to have walked this earth 200,000 years ago. So obviously something dramatic must have happened in the last 100 odd years to make this happen. Now, doing research in this area, you would actually realize there's some very fundamental things that have changed in the last 100 odd years. Number one, availability of potable drinking water. Number two, the importance of hygiene. Number three, housing. Number four, availability of food. 5, the ability for us as humans to take care of disasters. That list goes on. But on top of that list is the emergence of modern medicine. Okay. And to me, modern medicine has truly changed the curve of life. Now, if COVID has reduced the global average life expectancy by about a year and a half. It's on the upward trend again. But if we now fast forward another 100 years, our children and our grandchildren, how long will they live? Would life expectancy double? I mean, of course, it's for anyone on this audience to guess whether it will double. The fact of the matter is that the global population is aging. And the consequence of aging results in some very fundamental things. The impact on health, especially chronic diseases. and socio-economic factors which have to be considered. And therefore, I leave you with this first slide to think about it. Because if I have to ask any of you, including myself, do any one of you want to die? The answer is no. We want to live and live as long as possible. But there are consequences of living longer, and that's something we need to think about as a human race. Now, again, if you look at once modern medicine has come, there are thousands of drugs that have emerged in the market, some of which I believe are truly transformative. And if you notice on this slide, the center, I put insulin. And I've deliberately put insulin because I worked predominantly in diabetes research. But I believe that when insulin came in the market way back in the mid-1920s, it changed the way patients with type 1 diabetes were, and it saved lives of patients with type 1 diabetes because of the consequence of diabetic ketoacidosis, and they were dying young. But not only as insulin, you look at vaccines today, you look at antibiotics, to a large extent, we have conquered infectious diseases. Not necessarily in our own country, but as a race, we have conquered it, and we are almost there in countries like ours. But not only have we increased the number of years we live, I think in some cases, drugs have enabled us to live better lives. Let's take the example of the H2 receptor antagonist. Prior to the emergence of the H2 receptor antagonist, the line of therapy for peptic ulcer disease was surgery. H2 receptor antagonist changed that entire approach to make it medical line of management. And therefore, I believe going forward, there will be more transformative drugs that will emerge. And people like you who are there, who should make and bring these transformative drugs to man. Now, the question is, why is pharmacology important? And how does pharmacology play a role in drug development? The very essence of pharmacology are built on two pillars, kinetics and dynamics. And there's a close relationship between kinetics and dynamics. The concept of building a drug today is based on the principle of what is termed as a drug dose concentration effect relationship. So in other words, let us say in this particular flow diagram, you give a drug orally, you attain a desired concentration in the blood. The reason you need to attain a desired concentration so that you have a desired concentration at a target, and by securing a desired concentration at a target, you get an effect which then translates into a clinically meaningful outcome. Now, I want to bring out two very fundamental things that we learned in pharmacology, probably in our first or second class in pharmacology, undergraduate medicine. A drug needs to bind to a target, what we term as affinity, and needs to produce an effect, what we term as intrinsic activity. And therefore, pharmacologists like us should be able to determine the binding and intrinsic activity to be able to explicitly state the value of the drug way before it is actually reaching the market. Okay. And to a large extent, we can do that because this is our bread and butter. We've learned it way back in medical school. How does pharmacology play a role in fundamental biological research? To me, it plays a role end to end, beginning from the idea right up to the end of life cycle management. Let's take the example of targets. I did mention that when you want to influence a disease, you first and foremost need to know the natural history of a disease. Now, by knowing the natural history of a disease, you know what happens, you know what triggers the disease, you know what happens at a macro and microvascular level. If you know that, then you can potentially identify targets. And it is the role of the pharmacologist today to identify biological targets and newer targets and validate these targets. And what do I mean by validate these targets? I mean that show consistency in an effect and confirm to what extent by influencing this target you are impacting the disease. So in today's world, we not only identify new targets, I must admit, for certain diseases, we look at multiple targets. And why do we look at multiple targets? Because certain diseases have multi-dimensions and multiple pathways. And we now know that single drugs sometimes influence multiple targets, or a combination of drugs often influence multiple targets. The objective is to have a clinically meaningful outcome at the end, possibly as good as standard of care, if not better than standard of care. But one of the things that have recently emerged, I would say in the last 1 to 2 decades, is this whole construct of drug repurposing. When we as pharmacologists and our understanding of pharmacology understand how drugs work and understand by which pathways they work, we now know that diseases, even diseases across systems, sometimes have similar pathways. And therefore, today, if we have a drug which is indicated for a particular disease, we could potentially use that drug for another disease because it's likely to influence a pathway which is similar to the pathway and the indication that we've already secured. And that there has been a lot of focus on drug repurposing in the last decade or two. But in addition to the impact we have on targets, one of the things that pharmacologists often do is to support preclinical development. Now, obviously, before we move to man, you need to study it in tissues and you need to study it in whole animals, what we termed as in vitro and in vivo experiments. Today, over the years, there are certain animal models which are already validated for certain diseases. But our quest is always to improve the disease model. But not only to improve the disease model for already existing drugs and models, but predominantly to look at models and validate models when animal models do not exist. So this is, to a large extent, the role of a pharmacologist in preclinical development. Now, this presentation is to talk about the role of pharmacologist, and I'm summarizing here with respect to the impact that a pharmacologist has in a multidisciplinary team. I must admit that developing drugs require truly a multidisciplinary team. This team has knowledge, it has competencies and has skills. But to me, a pharmacologist to a large extent is more or less in the center of this team. And what does he or she need to do? He or she needs to communicate his or her knowledge to the other disciplines, but also understand what the other disciplines contribute to. I call it consolidation and therefore convergence of knowledge to be optimal for the project in question. But one of the things that you may need to do is also alter the manner in which you do certain scientific activities. And why is that? Because you need to align to what the project needs to do. Okay. So even though you may have some good ideas, sometimes you need to park your ideas and then navigate to provide your scientific inputs to projects. Last but not the least, one of the things that is your responsibility going forward is to collaborate and have communication and collaboration with both academic scientists and industry scientists. To me, knowledge resides in different locations. It is for you to seek that knowledge and bring updated knowledge to the table when you discuss it as a scientist in your development team? How does the role of a pharmacologist and pharmacology in exploratory development? In exploratory development, to a large extent, we address three pillars. One is the drug tolerable? In other words, can it be tolerated by a human being? What is the kinetics of the drug? Does it follow linear kinetics or first order kinetics? And at particular drug levels, does it induce a pharmacodynamic response? A pharmacodynamic response that you wish to see so that you can seek an indication in question that you are actually looking at. So some of the things that you do in exploratory development is, as I mentioned earlier, explore dose concentration effect relationship. But in addition to that, look at the shape and location of the dose concentration curve, because the shape and location of the dose concentration curve can contribute both to so-called desired and or undesired effects. And it's incumbent on you to be able to identify this, okay, and share it with the team. What about dose ranging, dose ranges? This is, again, fundamental in exploratory development. You need to identify a starting dose, dose adjustment, and an upper end of dose so that you're confident that I've tried the spectrum of doses which can potentially be used in the indication that you're exploring. Lastly, what you do in exploratory is you need to confirm proof of concept. And this is normally done in a phase 2A study. So the proof of concept is, does it work in the disease in question and at the doses that you have now tried? So when you look at exploratory development, you look at starting at first in human to proof of concept. And the pharmacologist, clinical pharmacologist, plays a significant role in determining this. And therefore, once you finish exploratory development, you often go to senior management in your organization and go for what is termed as go, no-go decisions. You need to have all the facts in place. You see if those facts meet what is termed as a target product profile. And if they do, you, to a large extent, have to support the progression of this drug into full development. Okay, that is the role that you have. Of course, you would be supported by your other team members. In addition, what you need to do in exploratory development is determine what is termed as the primary and secondary pharmacodynamic parameters. So what is this simplistically? The drug acts on the target, what is termed as the primary target, and you have to determine the dose concentration response as a consequence of binding to this target and target alone. But sometimes when you administer a drug, it could act on other receptors or targets which you're not aware of. Okay. The consequence of it binding, so-called off-target effects, can sometimes result favorably in desired effects, but more often than not in undesired effects. Now you have to explicitly find out at what doses are you having these so-called undesired effects. Are these effects permanent? Are they reversible? Are they dose related? Okay. And if for example, if there's a desired effect, what is the contribution of the desired effect versus the primary target? Clearly, as a pharmacologist and pharmacology, pharmacokinetics being one of the major pillars, plays a significant role in drug development. Let us say in your target product profile, the team has identified that this drug needs to be given once a day. Then it is incumbent on you to say that, okay, I believe this drug has an adequate half-life or long enough half-life to permit a once-a-day dosing. What happens if the drug has a low therapeutic index and doesn't have a constant bioavailability? Now you're in trouble because you may have spikes of safety issues and sometimes lack of efficacy. Okay, so this is something that you need to take care of. So you have to have constant bioavailability if you have a low therapeutic index drug. And if you do so, even in diseases like arrhythmias, you can potentially have an therapeutic drug, provided you can maintain your drug levels within the therapeutic window. One of the things that you need to understand early in development is how does the drug being handled by different species? Because if you now have concordance to a large extent between, let's say, a rodent species and a non-rodent species, the possibility of having concordance with humans is much higher. And this is something that you have to bear in mind. Does the drug have express genetic polymorphism? And what is the consequence of the genetic polymorphism? Does the drug inhibit or induce enzymes? And what is the consequence of inhibition and induction? The consequence can obviously be drug interactions, but can also be issues about efficacy of that own drug, safety of that own drug, and dosing of that drug. What happens in the course of drug development? You identify metabolites. What happens if it's an active metabolite? So what happens if it's a new metabolite you've never seen earlier? So if it's a new metabolite, you need to identify it. It will be characterized by a medicinal chemist. Okay. And you need to synthesize it and take it right through the whole preclinical development all over again. If it's an active metabolite, the question is, if you have an active metabolite which has a long half-life and longer than the parent compound, which is also active, then the question is, how do you administer this drug? Dosing may be a challenge. So these are things you ought to think about when you study the PK in drug development. When you do first in human studies, to a large extent, the clinical pharmacologist is the center of how first in human studies are done. And this is what a clinical pharmacologist would need to do. First and foremost, have repeated discussions with a toxicologist to understand the findings. Understand how these findings occurs and whether there's a cause-effect relationship. Now, if you understand this well, then you can proceed to look at humans. If you do not understand it well, or if the studies are inadequate, or if you feel you're not confident enough to move to man, ensure that you do other studies. The key is to be confident that when you now administer in man, you are minimizing the risk to the minimal, if not that no risk at all. So therefore, when it comes to the role of a toxicologist, the toxicologist would provide to you target organ toxicity, but you as a clinical pharmacologist would need to minimize the risk and avoid significant adverse effects based on what you are seeing. So therefore, prior to first in human, this is the list of things that you would do. Identify target organ toxicity, look at NOAEL, which is the no observed effect level, no observed adverse effect level, the toxicokinetics, both at Cmax and AUC, and the MABEL, what is the minimum acceptable biological level for biologics. Okay, these are fundamental for you to have prior to first in human dosing. Now, what are the factors that you need to take into consideration when you design a clinical study in exploratory development? Obviously, you've seen some things in preclinical, okay, and you've seen some degree of toxicity. That has to go into when you develop your selection criteria for a study population. So make it as homogeneous as possible to begin with. What are the tests that you need to do, especially tests to provide safety monitoring? What is your starting dose? What is your dose increment? What is the range of doses that you potentially test? What is your maximum exposure you do? And last but not the least, once you studied in patients, or once you have a model of disease in a healthy volunteer, what is the impact on a clinical endpoint that will confirm proof of concept? Once you have got all this data and now moved to your full development, what we term as phase 3A, these are the principal things that you need to follow when it comes to design of a study. Ensure that you have an adequate heterogeneity in your target population. Ensure that you have the appropriate and selected doses to confirm efficacy and safety. ensure that you have the right clinical endpoint or the right biological biomarker. Ensure that you have in your secondary objectives pharmacoeconomic parameters that help you eventually get market access and pricing. Ensure that you do the study in the major regions of the world. And when you do it in multicentric so that you get an appropriate sample of that country and do it in multiple countries. I can tell you when I used to head in my former organization, the rest of the world for development, we had this 1/3, 1/3, 1/3 principle. 1/3 North America, 1/3 Europe, and 1/3 rest of the world. But I can tell you that when I left that role and took another role, we were already doing about 40% in the rest of the world. And to a large extent, countries like India were contributing significant to those numbers. You need to ensure that you have the appropriate active comparator. And when you do not have an active comparator or there's no standard of care, you ensure that you need to have an appropriate placebo. Regulators expect us to have at least 2 pivotal studies. But let me tell you, being involved in diabetes research, in your full development, the minimum number of studies that you would probably need to do is 5, and it could be even up to 10. Just to give you a heads up on what the cost of these studies are, anywhere in the range of between $50 to $100 million. So you can imagine just for your development or full development, the amount of money, effort, time, and resources needed to prove that the drug works well. Sometimes you move studies later on when you don't have adequate data to be able to do it in this group. So if you're looking at a new drug, let's say in diabetes or a new insulin, whether you're looking at children with type 1 diabetes or type 2 diabetes, whether you're looking at the elderly, type 1 pregnancy, gestational diabetes, or whether you're looking at the insulin to look at antibody development, this is what you can do as part of a 3B or if not phase 4 program. In phase 4, there are 2 types of studies that you can do. It was talked about earlier. You can do a randomized controlled trial in any of the ones which are mentioned in 3B, or you can get real-world evidence data, which the earlier speaker so well elucidated to. You have to look at getting data of patients in the license indication. You can get comparisons with other drugs, especially when your development program does not include those other drugs. You get clinical effectiveness data in real life. As part of life cycle management, you can have line extensions, new combinations, new formulations. Ideally, pharmacoeconomics data is done in phase 4, where you do a superiority study of your drug versus standard of care, where you believe that in certain areas, your drug is better than standard of care. You can explore new indications based on the data, the real-world evidence data that's emerging. You can do studies in special populations. And last but not the least, in phase 4, you now know in a real-world setting how does the drug work. I want to tell you something about the modern approach to developing drugs. And I'll talk about, there are 4 things mentioned here, but for reasons of paucity of time, I'm going to limit it to just 2. The first is about GLP-1. How do you think about getting a new drug to the market? GLP-1, if you do research in it, was identified way back in 1987, if I recollect correctly. What happens with GLP-1? It's secreted by the L cells in the intestine. And when you have food, these L cells secrete GLP-1, and these GLP-1 molecules, they enter the enteroinsular axis, cause a release of insulin in a glucose-dependent manner. So now, this happens endogenously in our bodies. If theoretically, it was thought way back in the 90s, if you can inhibit the breakdown of GLP-1, you can potentially have an anti-diabetic drug. And that's how the DPP-4 inhibitors emerged. Or GLP-1, when you give it, if you notice you give it intravenously, it is very rapidly broken down by DPP-4 enzyme. If you modify the GLP-1 and make the body not recognize it as a true GLP-1, so-called GLP-1 analog, okay, you can mimic GLP-1 and cause insulin release in exactly how the body does it. And therefore, it can be a very interesting, that's how the GLP-1 analogs came into being, the exenatide and the liraglutide. Okay, this was theorized initially. And now we have both the DPP-4 inhibitors and the GLP-1 analogs. I want to talk about another very interesting thing that was brought up, I think, earlier by Dr. Nitya, that talked about carb therapy. Okay. I finished my MD in 83. And if at that time, if you had asked me, is there a possibility that we can cure advanced acute lymphoblastic leukemia? The answer I would have said, you're talking rubbish. Okay. Today, that has become a reality, that we can possibly cure advanced stages of acute lymphoblastic leukemia. I've seen patients who are on carb therapy in a drug that I talked about earlier, Kymriah. Now, what is the basis of this? When a patient has acute lymphoblastic leukemia to begin with, there's a trigger that triggers it, and we don't necessarily know the trigger. The body's immune response develops T cells. And these T killer cells try to kill the cancer cells. Unfortunately, the rate at which the cancer cells multiply far exceeds the immunological response. So there is imbalance in favor of the cancer cells. Now, hypothetically, if you now withdraw the T cells, which are the active T cells, you genetically modify them and you externally manufacture them in quantities far beyond what the body's cancer cells can be produced, and reintroduce them into the body, you alter the balance. And that is the principle on how CAR therapy came into existence. And that is the way forward on how we will be able to tackle a lot of cancers going forward. I wanted to state that because I now know it is a reality, even for advanced acute lymphoblastic leukemia. Dr. Nitya also talked about personalized medicine a bit. And I must admit, in the last 30 years after my MD, personalized medicine has made small steps. I truly believe that we are on the threshold of making personalized medicine a reality. Why? There is a reason. We now have identified the human genome. I think she talked significantly about genomics, okay, proteomics, etc. in her first presentation. Our understanding of the human genome is far better than what it was 30. We did not have any indication 30 years ago. Technology has advanced significantly. I used to lead a team in my former organization about AI and ML. Okay. I always thought that there was only natural intelligence. Today, I know that artificial intelligence can supersede natural intelligence. It's unbelievable. Okay. And we have big data. Large companies have big data, which is unimaginable about millions of data points. And why have I put these three things here? Because I have mentioned this earlier. In science, for great things to happen, there needs to be consolidation and there needs to be convergence. And that is what is happening. I am telling you, I was a lead in this AI and ML team. And we used to identify hypotheses that I couldn't even have potentially dreamt about. Because you now have all the data points necessary to arrive at a hypothesis. As medics, we arrive at hypotheses based on the principles of deductive logic. This goes far beyond that. Okay. And therefore, I truly believe in my own lifetime, personalized medicine will become a reality. Since I'm talking to postgrads, and we live in a knowledge-based economy today, I could just put one slide on the need for knowledge, skills, and competencies. Some of the things, all the things mentioned in this slide, I have done myself. Okay, and that's why I told you my journey has been fascinating. It can move into another couple of more slides. Okay. I do not want to go into every single aspect of this slide, but I'll talk about the last. The previous speaker talked about publishing. I had a boss very early in my career who said, if you do not publish, you will perish. Okay. I do not have too many presentations. I only have 30 or 32. Okay. But make it a point to publish. Because in your lifetime, if you do not publish at least once every year, you will become redundant over time. I have 2 more slides, Ashish. If you give me the time, I will be able to finish it. Because these are some take-home messages that I want all of you to look at. Can we eradicate hunger? And why do I bring this up? Because it's very close to my heart. Today, I read an article in 2019. This was a couple of years back. At that time, there were 190 million people in our country that went to bed hungry. Can you imagine? Can you imagine? Politicians have done nothing. And as scientists, we need to do. And why am I telling you this? I'm going to give you a point of view, which I want you to reflect on. If you look at human race, we have moved from being hunters to being gatherers. Okay. And when we are gatherers, as we are today, we grow food and we eat. We eat 3 meals a day, 2 to 3 meals a day. Do we need to eat 3 meals a day? We feel hungry. That's why we eat it. To be honest, I had lunch today. I wasn't hungry, but I still had lunch. Okay. What is the consequence of that? Okay. There's adequate food in my system, adequate glucose, but I'm still eating. The question is, over time and over centuries, because we have become gatherers, our liver today only has a capacity of 100 grams of glucose that it stores as in the form of glycogen. So when you go to bed in the night and you wake up in the morning 8 hours later, you feel hungry and I want to have breakfast, because that 100 grams of glycogen is reconverted to glucose to maintain blood glucose levels in the basal state. Now, my thought to you is, if you can increase the capacity of the liver from 100 grams to save 100 grams is nothing. Okay. To save 150 or if not double, you will eradicate hunger and you will get the Nobel Prize and you will get the Bharat Ratna in India. Okay. Because you have done something that nobody else has done. And it is possible for scientists to do so. Can we cure diabetes? I believe in my lifetime, we will cure diabetes. Why? It is only a matter of time that the adult stem cell, which produces the beta cell, is refined enough not to have noise. And once we have refined that to the extent that there is no contamination of the adult stem cell that produces the beta cell, we have cured diabetes. I am telling you, I am predicting that this will happen in my own lifetime. And my last slide, Ashish, my very smart professor of physiology, in my very first lecture, put this slide up on premium non-noceric. As doctors, he said, first, do no harm. When we develop drugs, every drug can potentially harm, but we always look at benefit and we weigh benefit versus harm. And as long as the benefit supersedes the harm for the indication in question, we have the right to produce that drug and support that drug entering the market. I want to end here. I hope I've kept you still awake at the graveyard shift of my presentation. And I'll be happy to take any questions.
All right, thank you so much, Dr. Julius. I mean, on behalf of core team for Pharmacy Year 2023 and the students out there, I would like to thank you for being with us today and taking us through all the aspects, critical aspects that are there in terms of role of pharmacology in drug development. I know we have overshooted some time, but at the same time, I realized that yes, this is something which was critical for students to know and understand. Thank you so much, sir, for being
Thank you,
with
Ashish.
us today. All right, I do have one, I know we do not have time, but I would require your quick comment on one
Sure.
thing, and which is also relevant to the students. So for MD pharmacology students, what diverse career paths are available within the realm of drug development and pharmacological research?
So Ashish, today, just to give you a heads up, right? My last company before I retired was at Novartis. So I was the I used to head a major team there, a global team. That's a development team. Okay. So that's not a commercial team. That's a development team. Not that I'm against commercial teams, but every single individual we took in that team were folks like you guys. Okay. And across development, you can have the core roles that medics choose, okay, are in drug safety, in clinical development, okay, in exploratory development, okay, obviously in kinetics, okay, in medical affairs, and we also have global medical affairs. Okay. That doesn't mean that you cannot fit into the other areas. You obviously can fit into regulatory affairs, you can fit into clinical operations, but you will be competing with, as a medic, you'll be competing with other folks there, the PhDs and the MPharms as well. So now, in addition to that, obviously, like folks like you, Ashish, work in business offices, okay, and become medical advisors, right? And as a medical advisor, you need to now liaise with, you do clinical development, but you do local support for your team as well. And you liaise with different groups, including your clinical operations group, including with your regulatory group, clinical quality groups. So I can tell you there are multiplicity of career paths that you can choose. Just to give you a heads up, when I joined my former company in 2012, we were 600 people. When I left the company, we were 7,000. Okay. And I strongly favored positions coming to India. Okay. Why? Because we have talent like you guys to make it available. Okay.
All right. Thank you so much, sir, for responding to that. And I think with that,
Thank
let
you.
us call it a day. I would like to thank all the students for staying with us enthusiastically. Please stay tuned for day 3 of PharmaSteer, in which we have already planned some interesting sessions around omni-channel dynamics and how this is going to reshape entire approach which we have today. And at the same time, we are also having a talk on future of medical affairs with practice change at a scale. So these are the interesting things which is planned on day 3. So stay tuned for that. And with that, I would also like to thank our speakers for the day for amazingly taking the team through each and every critical aspect that is there in place. And lastly, I would also like to thank Dr. Sandhya Kamath ma'am from GS and KEM Hospital, Dr. Rakhi ma'am, Dr. Amitos, Dr. Padmaja ma'am for being there with us and for all the guidance and support. Thank you so much, everyone.
Thank you, Ashish.
Thank you, Ashish. Thank you,
Ashish.
sir. Thank you, all the esteemed speakers. Thank you.
Thank you, Dr. Oz.
Thank you.